Hello everybody. Welcome to Chromatin-Con 2023, and the topic is going to be epigenetics of aging. Um, we're welcoming everybody. Thanks for joining us. I'm going to remind everybody that after each speaker, we'd like you to use the Q&A button on the bottom of your screens, and that will allow you to ask questions. That way, we'll allow 5 to 10 minutes after each to answer specific questions. If you have more general questions, we ask that you leave them for a little bit later. We're going to have a roundtable discussion where I will attempt to let some of the audience ask their own questions, and we'll see how that discussion works. So in that case, you would use the raise your hand button. But for now, please use Q&A, submit your questions there, and we'll try to get to them. So with that, I'm going to ask our moderator, Ashley Webb, to take over and introduce our first speaker. Thanks, Ashley.
All right, great. Thanks, Mason, and thanks to Peter Adams and the organizers for putting together a fabulous meeting for the next couple of days. I'm really looking forward to it. And thanks everyone for coming. So for today, I'll be the first speaker. And the title of my talk is Sex-Specific Mechanisms of Brain Aging. And I'm an associate professor at Brown University. So I will share my screen. How does that look? Good, thank you for the thumbs up. All right, so just a couple quick disclosures before I start. All right, so in my lab, we study the molecular mechanisms of brain aging and neurodegeneration. I think as many of you already know, aging is accompanied by a loss of cognitive function, and that includes decline in learning and memory. So this is very common as we age. And at the same time, aging is the greatest risk factor for a number of diseases, and that includes diseases of the brain, so neurodegenerative diseases such as Alzheimer's disease. So for example, before the age of about 65 or so, pretty much nobody gets Alzheimer's disease. But after that age, there's an increasing prevalence of Alzheimer's disease as well as other neurodegenerative diseases. Despite these well-known issues in aging, we really don't have a handle on the molecular mechanisms underlying brain aging and neurodegeneration. And so this is what my lab is interested in studying, with the idea is that if we can understand these mechanisms, then hopefully we can come up with ways to intervene and improve healthy aging of the brain. Now, a lot of the work that's been done, for example, in the context of Alzheimer's disease, has been looking at a part of the brain known as the hippocampus. And so this is a cross-section through a mouse brain. And the hippocampus, which is the center for learning and memory functions, is right up here in the mouse. But there's another region of the brain that we have become very interested in recently. We feel this is an area of the brain it's fairly understudied in the context of aging, and that's the hypothalamus. And so the hypothalamus has a number of diverse functions that are really critical for healthy aging. Many of those functions are listed here. And so all of these things tend to change with age, right? So we often undergo weight changes with age. The hypothalamus regulates body temperature. It's harder to regulate your body temperature with age. Hypothalamus is also critical in pituitary control, so it's the H in the HPA axis, as well as these other functions that are known to change with age. Lastly, at the bottom here, sleep-wake cycles, so our circadian rhythms and sleep in general, definitely altered with age. I think we all feel that as jet lag gets worse and worse with age. But again, really the mechanisms underlying these changes aren't well understood. And so just to give you an idea of where we are in the brain, this is a human brain here, and the hypothalamus is the area that's shown in blue. So it's basically like right over the roof of your mouth. And then in the mouse, where we're studying the hypothalamus, and the data that I'll show today are from the hypothalamus in the mouse, the region is circled down here. So it's down below the hippocampus. All right, so not only do all of the processes that the hippocampus modulates change with age. It's also true that there is more direct evidence that the hypothalamus itself can actually modulate lifespan. So these are just a few studies that have come out in the last decade or so. There's work from Dong Shankai's lab demonstrating this. So if you alter the NF-kappa B axis in cells within the hypothalamus, that can actually extend lifespan. There's been a lot of work from Shinamai's group at WashU, also implicating the hypothalamus and lifespan itself. So this is just one of the figures from the paper where they generated the mice known as the Brasto mice. And so these are mice that have increased sirtuin and SIRT1 in particular activity in a couple of regions of the hypothalamus. And those mice have an extension in their lifespans. And then down below, lastly, there's a whole body of research from Andre Bartkey's lab implicating the growth hormone axis, including neurons in the hypothalamus, in aging and longevity. And so this is just one particular knockout mouse with an alteration in the hypothalamus. And these mice actually have you can see a pretty significant increase in lifespan compared to the normal controls in blue. Okay, so hypothalamic functions change with age. The hypothalamus itself seems to be modulating aging and healthy longevity in some way, yet the molecular mechanisms responsible are not very well defined at this point. And so this is a question that two of my graduate students wanted to take on starting a few years ago. It's really a new area in my lab. So these are both brave graduate students, Caitlin Hadanovic, who's a neuroscience graduate student here at Brown, and then Dodo Yu, who's an MCB graduate student at Brown. They're both Carney Fellows, so they're both fellows of the Neuroscience Institute here at Brown, and Dodo is also a Data Science Master's Fellow. All right, so what we wanted to do to begin was just really sort of a very simple experiment. So we simply asked, okay, how does gene expression change in the hypothalamus at the single cell level? So we wanted to look across all of the different cell types in the hypothalamus, and we compared young mice and aged mice at 3 and 24 months. We basically just dissected out the whole hypothalamus in these mammals, and then we isolated nuclei for sequencing. We had to use nuclei in this case because in the case of the brain, if you dissociate and try to take out and retrieve single cells, most of the neurons will not survive that process because the neurons are so fragile. So instead, we sequenced RNA from the nuclei. And we sequenced about 40,000 nuclei using the 10x platform. And this is just a UMAP, kind of give you an overall view of what these data look like. So on these UMAPs, I think most people are familiar, but remember that every dot represents a cell, or in our case, a nucleus. And we were able to obtain large numbers of neurons in our experiment. So those are shown in green, as well as the other major cell types in the hypothalamus. So for example, we have astrocytes, oligodendrocytes, microglia, even tanycytes, which are rare cells in the hypothalamus. So we felt pretty good about our data set. And without getting into too much detail, within just the neurons, we were able to identify 35 different subtypes of neurons. And those are shown here in this dendrogram. And we could identify, for the most part, the identity of these different types of neurons, as well as a major neuropeptide that's secreted from them in the cases where there's a major neuropeptide that's generated from the cell type. We also took advantage of spatial data that was available through the Allen Brain Atlas and projected our data sets onto their spatial data. And this was really just sort of a validation to validate the cell types that we pulled out. So for example, the astrocytes are shown here, and they're pretty much distributed throughout the hypothalamus. Whereas in the case of specific types of neurons, they were more localized within the hypothalamus, which is what we expected. And so just highlight a couple of examples here. CAR-T neurons are up here, and then the POMC neurons, which are found in the arcuate nucleus, they're feeding neurons. They are these bright yellow neurons down here at the base of the hypothalamus. So we felt pretty confident about the identities of our neurons that we were able to find in the hypothalamus. All right, so the big question that we had is what happens with age? So a few sort of points to give you an idea of the quality of the data set. The first key point is that we did not see major changes in the composition of the hypothalamus in our data sets with age. So this is just a UMAP where I've colored the cells either blue or red for young and aged. And you can see that each major cluster in the UMAP contains both ages, and so we didn't derive completely new clusters from the aging data set, and this was sort of a good thing, because it is known that the brain doesn't change majorly in composition with age, at least not with non-pathological aging. There isn't a lot of cellular death with age, and so it gave us good confidence that there wasn't something technically wrong with our data set, particularly with age. We were able to get back the cell types that we expected. And on the right, this is just another way of depicting this quality control measure. We had 4, I didn't mention this, but we did 4 replicates per age, so 4 young and 4 aged animals. All right, so importantly, we did pick up cell type specific changes in gene expression with age. I think this is generally sort of expected. So in this strip plot here, every dot now represents a gene, and any dot that's in color is representing a statistically significant change with age. So for example, astrocytes here had lots of changes with age. The neurons are down here in green, also lots of changes, but different from the astrocytes. And then there were other cell types that didn't seem to have as many changes with age, though in a lot of cases, we didn't have a lot of cells for that cell type, so it was a little bit harder to pick up significant changes with age due to low cell numbers. All right, then just a little bit about the types of pathways that are altered with age, and these were very cell type specific in nature. So for example, in the astrocytes, we saw a lot of changes in cholesterol homeostasis. We also saw alterations in mTOR signaling genes, and those changes in the TOR pathway genes were also found in the neurons, but then neurons also had other distinct changes with age, distinct from other cell types. In microglia, for example, gave us lots of changes in immune pathways and inflammatory pathways. So somewhat expected, but new insight, I think, into how these different cell types are altered with age. Now, just zooming in on the neurons were quite interesting. So I took off, just for simplicity, I took off the names of the different neuronal subtypes, but those 35 clusters that I showed earlier, those are on the left side over here. So each row is a different type of neuron. And then I've also taken off the names of the molecular processes that were altered with age, just for simplicity. But this is a gene set enrichment analysis. And so a few points here. First of all, we found that the different types of neurons were aging quite differently, and some of the neuron types had lots and lots of changes with age across many different molecular processes, whereas other types of neurons had relatively few changes with age, possibly indicating that they're more resilient to the aging process. And then the second point is that when we look at the molecular processes, there are some processes that are changed across almost every type of neuron, whereas other molecular processes seem to be sort of specific to one type of neuron. And we looked a little bit more deeply into the processes that are altered across many different cell types. What we found was pretty interesting, I think, which was that these hallmarks and pathways tend to be neurodegeneration associated pathways. So this suggests that potentially the gene expression changes that we've picked up with age here are the gene expression changes that make neurons susceptible to neurodegenerative diseases. So we're very interested in digging deeper into these sort of common mechanisms of neuronal aging, as well as the more resilient mechanisms. All right, so we wanted to ask whether we could observe trajectories of aging across these different cell types in the hypothalamus. So we ran some pseudotime analyses across our different types of cells in the hypothalamus. And interestingly, in most cases, we couldn't define a specific trajectory across any particular subtype. The exception of this was the macrophages and the microglia. And the microglia gave us a very interesting age-associated trajectory. So just briefly, the way that this works, these pseudotime methods were developed mostly for developmental data sets. And so, for example, here, if you have a developmental data set or even an aging data set that represents physical time, that's your starting point. And then if you take away all of the aging information or time information, and then just look at the the transcriptome information across each cell. That's what's shown here in B. So this is sort of a loss of temporal information that's been removed. You can apply these algorithms, these pseudotime algorithms, that will then reorder the cells across time. And that new time is known as pseudotime because it's learned from the data set. It's not real time. But in general, what the algorithm is able to do is reorder the cell in a way that represents physical time. And then once you have this pseudotime in place, you can then dig in and ask, okay, what are the particular gene changes that are changing across pseudotime and that are representing aging in our case of the cell. All right, so when we ran this analysis, this monocle analysis on the microglia in our data set, we got a very nice aging trajectory. And so this is the aging trajectory here. On the bottom, this is the representation of pseudotime. And so young pseudotime is on the left in purple, and then moving across to the yellow, this is going across pseudotime into aging. And then when we project the real-time information back on that pseudotime that we obtained from the algorithm, you can see that the young cells, interestingly, they cluster early in pseudotime, and then lots of aged cells are found later in pseudotime. So it does seem to represent an aging trajectory. But I will also point out that something that we didn't expect to see in this analysis was that some of the aged cells are actually still found earlier in pseudotime, suggesting that at least some of the microglia are maintaining younger features of a microglial state, of a more youthful microglial state, even in the aged brain. So we're very interested in digging into the differences between the young and the aged microglia within the old brain to see if we can understand better how to take those old microglia and bring them back to a more youthful state. And so we've dug a little bit deeper into these signatures across pseudotime. And one thing we did was we took some signatures that were developed by Ido Amit's lab. And these are signatures of either healthy homeostatic microglia, what's known as stage 1 DAM microglia. So these are basically aging microglia, or a third type of microglia. And these are microglia that are undergoing neurodegeneration. So for example, isolated from Alzheimer's brains. So we basically asked, does our pseudotime data set correlate with any of these different signatures of microglial states? And what we found, I think, was pretty interesting. So what we found is that across pseudotime, so again, this is our pseudotime data here, we see the peak of homeostatic microglia pretty early and into the mid stages of pseudotime, whereas homeostatic microglial signatures are gone later in pseudotime. Whereas the age-associated microglia, these DAM1 microglia, these are low early in pseudotime, and then they track upwards across pseudotime, so they become more highly represented later in pseudotime. And then we never really saw much enrichment for DAM2 microglial signatures in our data set. And this actually makes some sense because the DAM2 microglial signatures, remember, are representing a disease state. And our mice were just undergoing healthy aging, not neurodegeneration. We could then ask what specific genes were represented across pseudotime in our data set. And so we could divide pseudotime into 4 distinct modules, which is shown here in the heat map on the left. So module 1 is represented early in pseudotime, followed by genes that come up in module 2, and then 3, and then finally module 4 represents late pseudotime. And I realize that these, the text on this slide here on the right is a little bit small, but I'll just make a couple of general points. As we look across the modules and across pseudotime, initially we see signatures represented that are more representative homeostatic signatures, such as gene regulation and cell signaling, sort of normal functions of the microglia. But then as the cells progress through age, they then have altered signatures that are related to metabolism and organelle homeostasis, followed by an induction of cellular stress, and then finally alterations in quality control, trafficking, and viral processing. And I'll just point out the very last signature that's shown here is regulation of amyloid beta clearance. So it does seem that as these cells get really, really late in pseudotime, they are starting to represent more of a neurodegenerative state. Okay, so a couple of points about the extent to which these signatures that we pulled out are shared across brain regions. So here we're really focused on the hypothalamus, But as I mentioned, there's been a lot of work on the hippocampus, which degenerates very significantly in diseases such as Alzheimer's disease. And so we wanted to ask whether our signatures in the hypothalamus were shared in that brain region as well. And we were fortunate that Nicola Neretti's lab, who's also here at Brown, had done a very similar experiment where they did a single nucleus sequencing of the hippocampus using the same platform. So we basically took our data set and correlated it with theirs. And so we started with the glial cells in the brain. And what we found was that all of the different types of glia that we pulled out in the data set, so oligodendrocytes, microglia, as well as astrocytes, these tended to be correlated across the different brain regions. So at least at the transcriptional level, these cell types seem to have similar signatures of aging. But what was interesting was when we looked at the neurons, we saw very different signatures of aging, suggesting that the neurons in the hippocampus are aging in a very different manner from the neurons in the hypothalamus. And this is potentially very interesting in the sense that it could give us some insight into why hippocampal neurons are more susceptible to neurodegenerative disease compared to hypothalamic neurons. So we're digging into this a little bit more in the future. All right, so in the last few minutes, I want to tell you about a feature of hypothalamic aging that we pulled out that was a little bit of a surprise in our data set. And what I didn't tell you at the beginning was that the way that we set this experiment up was actually only using female mice. And so we think it's really important to understand both male aging as well as female aging. And at this point in time, when we started these experiments, we thought, okay, we need to just focus on one. We couldn't do both sexes. So we started with female mice. And the reasons that we want to look at both female mice as well as differences between males and females are several. So first of all, it's well known now that lifespan is sexually dimorphic in most species. So in humans, females tend to live longer, and that's true in a lot of mammals, but across other species, there are other cases. So in some species, males actually tend to live longer. And in addition to that, we know that some diseases disproportionately affect one sex. So for example, heart disease is more prevalent in men. In women, we actually get more Alzheimer's disease. That's shown here in this diagram, right? So about 2/3 of people who have Alzheimer's disease right now are women. We don't understand the molecular basis for that at all. There are few studies that actually address the mechanisms of female aging at this point. And so we felt that this was a good time to start doing that and to also start comparing males and females. And then lastly, many interventions that extend lifespan affect only females, right? And so, so we don't understand these differences, again, between why females sometimes respond better to certain interventions and why males respond better to others. But we need to start sorting this out, in my view. Okay, so we just looked at females in this particular study, and that led us to a very unexpected finding. So when we looked at gene expression changes just in the neurons with age, we found that the top most upregulated gene was a gene known as Exist. And this is a gene that is expressed on the X chromosome, and it's a key modulator and sort of the master regulator of X chromosome inactivation. And this wasn't just true in a few different types of neurons. This was pretty uniformly true across all of our different types of neurons. So this is back to those 35 different types of neurons in the hypothalamus that we found. And the Exist is labeled where it's differentially expressed in the different types of neurons. We also found upregulation of a related gene known as SciX, shown here, which is also part of the X chromosome, which is known as the chromosome inactivation center. All right, so just as a reminder, in terms of how the biology works in humans, so males are XY, females are XX, and in order to achieve dosage compensation, females express this Exist RNA just from one of the X chromosomes. And that happens during development. So once the Exist RNA comes up, it actually coats just that X chromosome that it's expressed from, and then it recruits epigenetic modifiers that then silence the X chromosome. And so once that X chromosome is transcriptionally silenced, it's actually sort of set aside in the periphery of the nucleus, as shown down here, and then it remains transcriptionally inactive for the most part throughout life. So this is the process of X inactivation. Again, this happens early in development, and it's thought to be stable across lifespan once it occurs. So this was sort of strange that we pulled out this modulator of X chromosome inactivation that's a developmental modulator. So the first thing that we wanted to do was just validate this finding. And we did that in a few different ways. One thing we did was we went in and we did RNA scope for exist on the hypothalamus, and we looked across a number of different animals, and we found that indeed, when we used this method, there was an upregulation of exist in the hypothalamus. We also checked by RT-qPCR. Here we're looking at a few different brain regions from the female brain, and we actually saw a bit of a trend in every brain region, though in this case we saw the strongest upregulation in the hypothalamus. Now, importantly, we did not see major changes in exist in the male hypothalamus, and so that's an important finding because it suggests that this may actually be a female-specific phenomenon. So this is actually data down here on the right. These lines, there was just no detection. We also were very curious to know whether this might be happening in the human brain. It was a little bit difficult to find studies that included females, but in the case of Alzheimer's studies, there were a lot of female data sets out there, in part, I think, because there are more females who get Alzheimer's. And so we looked at a couple different data sets that were publicly available. They looked at 2 different regions of the brain. And when we specifically looked at exist expression in these different data sets, we found that indeed in the entorhinal cortex, there was an upregulation of exist in Alzheimer's patients post-mortem. And this is an important area of the brain that projects into the hippocampus, and it's actually an early area of the brain that degenerates in Alzheimer's disease. We did not see this change in the prefrontal cortex, and this is an area of the brain that is affected in Alzheimer's disease, but a little bit later in the disease. So these are some initial findings that indicate that possibly, at least in the context of neurodegeneration, there are changes in exist levels. So this biology, something might be going on with X chromosome biology in females as well in age-associated diseases. Okay, so this led us to question whether exist might be a biomarker of aging. So to answer this question, we turned to a machine learning approach, and we moved back to our own data set, and we just took all of the genes on the X chromosome as input, and we looked only in the neurons because this is where we saw this change most strongly, and we fed those data into a few different actually 8 different machine learning algorithms, and asked if we could identify algorithms that could then predict the age of neurons. So could we correctly identify a neuron as either a young, healthy neuron or an aged neuron just using genes and features on the X chromosome? So this is just the run through of the 8 different machine learning algorithms that we tested. XGBoost tended to be the best performer on our data set. And so we ran XGBoost using a number of trials on our test data set. And we found that about 85% of the time we could correctly predict an aged neuron as aged, and about 67% of the time we could predict a young neuron as young. So for machine learning, that's pretty good performance. We were pretty happy with that prediction. We then dug into sort of looking under the hood and this machine learning algorithm, what's driving the algorithm and allowing it to predict the age of a cell. And so the way to do that is to basically go in and start removing features one by one and ask whether the performance of the algorithm is compromised. So does the algorithm break when you start taking out certain genes? And so that's what's shown here. So this is the performance after removal of these different features. And interestingly, the feature that seems to be most important for this to run is exist expression. So if we took out exist, then we lost performance in the XGBoost algorithm. There were other features that also contributed as well. So what we really want to know, of course, is what are the functional consequences of elevated exist with age? Why is this happening? Is this good for the cell? Is this bad for the cell? Is this just neutral? And I don't have an answer to that today, but that's sort of where we're going with this in the future. Okay, so just to sum up and give you an idea of some of our future directions, today I told you that we have generated basically an atlas of the aging hypothalamus, and we see distinct transcriptional signatures of aging in different cell types and lots of distinct differences between different types of neurons. We also found a female-specific feature of neuronal aging, and that's elevated expression of the X inactivation center with age. And we also find this feature in the Alzheimer's disease female brain compared to age-matched controls. We were then able to apply a machine learning algorithm and show that EXIST can be a predictor of neuronal age in our data set. So again, we have a number of outstanding questions here. So what are the mechanisms that are underlying EXIST dysregulation in neurons? Does this actually impact cellular age when EXIST is changed? We're also very interested in understanding whether there are sex-specific epigenetic changes on sex chromosomes with age. So far we've only looked at females. What's happening in males? Males also have an X chromosome. And then lastly, we're very curious to look into whether the hypothalamus is affected in Alzheimer's disease, because many of the functions of the hypothalamus, such as regulation of sleep and circadian rhythms, are well known to be affected in Alzheimer's disease as well. Yet we know almost nothing about how this part of the brain changes in neurodegeneration. All right, so I will stop there and I can take questions. I'll just acknowledge my wonderful lab here at Brown. And I already mentioned the main drivers of this project, Caitlin Hadanovic and Dodo Yu, with a lot of help also from Lexi Hassel, my research assistant. And I will also acknowledge funding for this work, including a new NSF center to look at sex differences in aging. And I'm happy to take questions. All right, so I will stop sharing. Vera, do you want to, you can unmute, go ahead.
Okay, well, amazing story, Ashley. I'm just very curious of the continuation. So a couple of questions. So what actually happens to X chromosome? Does it, is it more activated or more silenced with aging?
Great question. So what we think is happening, and this is a lot of hand waving at this point, is still a model, but what we think is happening is the X chromosome genes overall are elevated with age, and what EXIST is doing is it may be coming up to try to get the chromosome back to homeostasis, to try to bring the level of the genes back down. But that's really just the correlative nature that we can, or the correlative data that we have right now. It's not functional data yet. So we're working on, for example, designing CRISPR systems to alter EXIST activation itself in an acute way to see if we can actually get at the mechanism a little bit better.
And one more quick question. So I saw one of the changes that you noted was in aged neurons was viral processing. So I wonder if you can comment on that, or if it has anything to do with our beloved transport.
Yeah, no, it's a great question. We haven't done anything to look more specifically at that, but I know your work and the work of John Sedeby here at Brown, I think there's a good chance that there's something going on there, but we haven't looked in detail. Yeah, thanks. All right, Raul, I see you have your hand up as well.
Yes, fantastic story. Actually, I have also a couple of questions. The first, related to EXIST also, that the RNA scope has enough resolution that you could see whether it's expressed on the second copy or if it just increased expression on the inactive X.
Yeah, also a really good question that we don't have the answer to. And the reason we don't know is because this was done on inbred mice. And so we can't tell the difference in our transcriptome between the active and the inactive X. And we didn't know we were going to find this. So
I know you cannot separate the two by genetic, but I thought whether the RNA scope, the resolution is enough that you can see the two dots in each nucleus or only one dot, one cloud for the X's. That was my.
Yep. So good point. So we haven't noticed a second cloud so far, but as I'm sure you know, the resolution of the RNA scope isn't that great. So we're actually going in with higher resolution methods. And the idea is to stain both the X chromosome itself as well as the EXIST, so we can really, truly co-localize, because I don't feel like we have the power yet to really answer that question with confidence. But it's an important one.
Very quick second question. Yeah. I noticed in the change of genes in neurons, MYC and G2M, which are markers of proliferation. You have proliferating neurons in enough frequency in the hypothalamus that will drive this signature change in aging?
Yeah, so that was a really interesting signature. And again, I don't have functional data supporting this, but I will mention that, so in the case of Alzheimer's disease, as neurons age and they begin to undergo neurodegeneration, a feature of that is that the cells can sometimes try to enter the cell cycle. And so in Alzheimer's disease studies, people have picked up those signatures pretty frequently, as well as the TOR signature, and all related to each other. And so, at least in the case of neurodegeneration, that is thought to be something that kind of puts cells into crisis and actually causes cellular death. I didn't actually expect to pick it up in normal aging, but it may actually suggest that these neurons are sort of primed already in normal aging for that feature. And then what happens is in Alzheimer's disease is something triggers it even further. But I thought those were really interesting signatures as well, and a little bit surprising, but maybe not so much in light of the Alzheimer's disease now that we know. Yeah. All right. So checking the time. Okay, so we have a couple more minutes, so I can answer a couple of questions from the chat.
Yeah, Ashley, if you'd like, I can read them to you.
Sure,
If
yeah.
that helps, or okay.
That'd be great.
So I'll just start with some of the first ones. So this was early in your talk. Reinhard Stoger asked, may have missed this, but was the single cell RNA-seq done on both sexes?
Yeah, so I hope I addressed that, but I will say that we have already obtained the data now on males. So we've basically redone the whole experiment, comparing directly males and females. We're looking at an intermediate age as well, and we actually have a multiome data that also includes ATAC-seq, because we really want to understand the epigenetic state of the chromosomes alongside the gene expression patterns that change with age. So stay tuned, it's coming, but we're just analyzing those data sets. But I will say that we haven't picked up so far, again, in that data set, exist did not change in males.
Let's see. So, was there any specific mouse strain that you used in the study?
Yeah, so these were C57 black 6 mice. So pretty standard strain. It's the most commonly used lab strain in our lab. They're great for things like behavior.
Umet Gerlovic asks, in your first analysis, why did you focus on transcripts only in the nuclei? I think he means for the single cell RNA-seq data, instead of the transcripts in the whole cell.
Yeah, so we would love to do whole cell analysis, but the issue with the brain is that the dissociation actually kills most of the neurons. And so we've actually tried it with whole cells in a couple of different brain regions, and you can get really great data back on oligodendrocytes, OPCs, different types of glia, but you just don't get neurons back. And so that's the reason. Although it does come with some limitations, we realize, doing the nuclei.
Next question is, what is the contribution of age-related increase in neuronal polypolarization for exist expression? And One Ronde says, thank you kindly.
We have not looked at that. So it's polycomb or polyploid?
A polyploidization. Yeah.
Oh, we haven't seen that so far, but we also have not. We haven't really applied the right tools to directly address that question. But yeah, good question.
So I'm not sure who asked this question, but they ask, have you tried looking at GnRH analogs or antagonists together with exposure to estrogens to see if you could manipulate the mice that way?
Yeah, no, that's a great question. And I think that this study has really raised a lot of possibilities in terms of interventions that we could use related to hormones, related to sex hormones, growth hormone. So I think we have lots of ideas there, but we haven't yet directly addressed any of those questions and how this part of the brain would be altered under different conditions.
And I think we might have time for another one or two. Mare Doyle asks, do you know whether exist is elevated with age in any other tissues?
So I've sort of heard offline just from sort of discussions, informal discussions from other people, but I'm not aware of there are publicly available data out there that if you mine them, you can find it, like in these Alzheimer's data sets. But for the most part, I have not seen that published from other groups. I think in part, again, because historically, like our lab as well as other labs, we tend to look at males, right? And there are a number of reasons for that. And I think it's just now that more female data sets are coming out. So I think we'll know soon, but it's not published yet.
Great. Let's see. I'll take one last one.
Mhm.
Robert Clark asks, overexpression of exist has been associated with affective disorders in females. Is that the same mechanism, you think?
I don't know, but I think that's a really interesting question, and I think that even more broadly, I think that there's a lot to learn from neurodevelopmental disorders, which disproportionately affect males. And the reason for that is that there are a lot of brain-associated genes, or like synaptic genes, for example, on the X chromosome. And males, of course, are more often affected because they're XY, and so you only need one copy of a mutation in order to get the disorder, whereas females tend to be, they're mosaic, and so they may or may not have the disorder. So it's a great, I don't know what the relationship is, and there isn't a lot of sequencing data on those cases right now. So I think that, in the coming years, I hope to get a better handle on that and the relationship between what's going on in our data set and those disorders. I think that's a really interesting thing to think about.
Yeah. So we have a lot of extra questions for you, Ashley, but I think we're going to run out of time. So we'll save those questions either for the roundtable, or if you like, we can address these. Either you can type into the Q&A chat box to answer them, or I can send you them later and you can answer them offline.
Okay, great, thanks. And we'll see what we can get to during the discussion, because we have some time at the end.
Yeah, perfect. But I'm guessing other speakers will have lots of questions as well. So, yeah, I'll let you move along.
Yeah, great. Thanks so much, and
Thank
thanks
you
for helping to keep us on time there. So next up, we have Raul Mostoslovsky. All right, from Harvard, and that looks good. All right, so his title is Linking Aging and Metabolism to Epigenetics in Cancer, One Gene at a Time. All right, take it away. Looks good.
Thanks, Ashley. Can you hear me well?
Yep, sound great.
Okay, so yeah, thank you. Thanks. I wanted to thank the organizers first, Peter Adams, and all the team at Motif for inviting me to be here today. I wish I could be in person there in California. Last time I went to the West Coast, I was lucky to go to the Grand Canyon for one night, and I took this picture that you see here, which actually was selected for Photo of the Day, National Geographic, and got an award from Nature Photographer of the Year. So very proud of the picture. Hopefully, at the end of the meeting, I can tell you how I took it. But moving on to the science. So my laboratory has been very interested in understanding this crosstalk between metabolism and epigenetics for many years. And I call it here an obvious link. Although many people in the chromatin field don't think about metabolism, and vice versa, many people studying metabolism don't think about the epigenome. But I said an obvious link because, and you can see here a few examples, for most of the chromatin modifications that we have in our cells, you need specific metabolites, and they tend to be limiting. You cannot methylate histones or DNA if you don't have enough amounts of SAM, the S-adenosine methionine universal donor. The same is true for acetylation of histones. You need acetyl-CoA, a major nodal point in the mitochondrial metabolism, and actually work from many, including Shelley Berger, show nicely, and Cathy Wellen, that if you reduce levels of acetyl-CoA in hours, you see changes in histone acetylation. So availability of these metabolites are key for these epigenetic changes. And lastly, they are not only substrate for the reaction, they are also key cofactors for many chromatin factors. And a good example is NAD, one of the major molecules in the TCA cycle in the mitochondria, that is used as a key cofactor by many chromatin enzymes, including PARPs and sirtuins. And my lab has been working on one of these sirtuins, SIR6. I don't think I need to introduce sirtuins in this audience. Originally described in yeast, they all use NAD as a cofactor, as I mentioned, and many of them extend lifespan. My laboratory has not been working on aging, but I believe I am being invited to this meeting because we've been working on one of these sirtuin called SIR6 in mammalian cells. It's one of the exclusively found primarily on chromatin. That's why our interest is coming from the epigenetics field. But interesting to say is that when in mammals, one of the main mammalian homologs that did show extension of lifespan is SIR6 itself. And recent work by Vera Gorbunova, who will be a speaker later in this meeting, showed that the activity of CIR6 is linked to longer living species. So as I said, we haven't been working on aging per se, but it's a very unique molecule to study. I won't have time to tell you about all the work that we've been doing in the lab, and we move away from only CIR6 in the past few years. We've been interested in understanding roles that is unique, the acetylase, and we've been publishing throughout the years major roles for CIR6 in regulation of tumorogenesis, and this will be one thing I will tell you about today. But we've been expanding, as I said, to other roles of metabolism in controlling changes in chromatin, particularly roles for one-carbon metabolism that I'm not going to tell you today. I keep my close interest in roles for chromatin in DNA repair, and we've been working on this in the lab also, but as I said, I won't have time to tell you about it today. And more recently, we became interested in unique adaptation of metastatic cells compared to primary tumors, and I hope I will have time to tell you one story related to these last studies. So the two stories I hope to tell you today, one is relates to metabolic heterogeneity in cancer, and the second one relates to non-genetic drivers of metastatic disease. And for the first, it's based on our original work with CIR6. I won't show you the original data. This we published many years ago, but we discovered CIR6 as a key modulator of what we call a glycolytic switch. Most of the normal cells keep CIR6 sitting on genes, on metabolic genes, co-repressing a factor called HIF-1 alpha. And this is true in normal oxygen and nutrient conditions. By sitting there, it's repressed HIF, so you don't get expression of what are called glycolytic genes. And here are a few examples: pyruvate dehydrogenase kinase 1, lactate dehydrogenase, and the glucose transporter GLUT1. By keeping repressed those genes, most of the pyruvate that get into the cells go to the mitochondria to produce ATP through respiration, the most efficient way to produce ATP from pyruvate. Conditions of low oxygen or low nutrients, we found that CIR6 is kicked out from chromatin. You transactivate HIF-1 alpha, you bring co-transcriptional activators that they acetylate these genes, and by acetylating these genes, we get upregulation of these genes and a switch where the cells, instead of respiring actively, they keep respiration, but in much lower way, but they start producing much more lactate from pyruvate. Less efficient to produce ATP, but gives the cell an opportunity to survive until conditions improve. We showed this in normal cells many years ago, and it was one of the first examples of a metabolic switch that was controlled by a chromatin regulator. Now we realized that, and I'm sorry, a sidetrack story, one slide. When I originally created this figure, this animation, we put CIR6 at the promoter, because most of the HDACs, histone deacetylases, repress transcription by sitting on the promoter, compacting chromatin, and inhibiting the recruitment of the polymerase. This original model was wrong. Work we did on ChIP-seq to see where CIR6 was sitting genome-wide, and show us that different from instant for HDAC1. Here you have a heat map where you see binding of the gene, of the proteins on genes. Each lane is a gene, and you clearly see HDAC1 sitting at the TSS. You're looking here at the TSS, 3 kb upstream and downstream. And in contrast to other HDACs, we see CIR6 sitting intragenically. And long story short, what we found in this paper was that it's a very unique HDAC in the sense that it binds the polymerase, move intragenically, and control what is called transcriptionally pausing and elongation by regulating the recruitment of specific elongation factors that I will not have time to tell you today, but please refer you to the paper. And we believe it's a unique adaptation of this HDAC to regulate and fine-tune expression of metabolic genes. And we can discuss this at the end if we have time. But back to the metabolic story, what I didn't tell you is that by getting rid of this transcriptional co-repressor CIR6 and HDACs, we were eliminating the sensing of these changes in nutrient or oxygen. And our cells that didn't have CIR6 were constitutively producing lactate, regardless of the levels of oxygen or nutrients. And this is called aerobic glycolysis, and it's a feature that Otto Warburg described many years ago in cancer cells. And Warburg said, regardless of the amount of oxygen, cancer cells tend to produce lactate from glucose. He didn't understand exactly why, but this was exactly the phenotype we saw for these CYP6 deficient cells. So we hypothesized that maybe cancer cells get rid of CYP6 to acquire a metabolic adaptation. Now, for the past 10 to 15 years, many laboratories have been studying this Warburg effect in cancer, trying to understand why cancer cells do Warburg. And this is a very heavy slide. I'm not going to go through all the details. The only thing I wanted to mention here, that from the work of many laboratories, what we believe is happening is that during the process of converting pyruvate into lactate here, instead of sending pyruvate to the mitochondria, which is a much more efficient process, you accumulate intermediates. And these intermediates are used for side metabolic reactions, particularly the generation of nucleotides, RNA and DNA nucleotides through the pentose phosphate shunt, acetyl-CoA for lipid synthesis, and serine through glycerol 3 phosphate for amino acid synthesis. And the production of the three basically lead to biomass, to an increase in biomass, and rapidly proliferating cancer cells may benefit from doing Warburg by obtaining these bricks for biomass. At the same time, in every one of these step reactions, you produce reduced NADP, contributing to a major change in reductive power. So the field has been thinking that the major reason you do Warburg is to produce bricks for biomass and to increase reductive power in these cancer cells. So many laboratories have been studying the why, no one really understood the how. And it was accepted that some of these cancer cells randomly acquired this metabolic adaptation, and these were selected for. Our results suggested that this may be driven by an epigenetic change. So we decided to investigate that hypothesis that maybe tumor cells get rid of CYP6 to acquire this metabolic advantage. In other words, CYP6 may be acting as a tumor suppressor. And long story short, this is exactly the case. We've been publishing on many different models. Here are two colorectal cancer and pancreatic cancer models of cancer. The moment we delete CYP6, the animals acquire much more tumors, bigger tumors, and in the context of colorectal cancer, we converted normal benign adenomas into aggressive infiltrating adenocarcinomas. In the context of pancreatic cancer, we make the disease highly metastatic, something that you don't see in the classical models of pancreatic cancer. So getting rid of this chromatin factor made those tumors extremely more aggressive, and we believe this is true because of a driving in this metabolic change, making those tumors much more glycolytic. Now, we published this few years back. The question that intrigued us at that point, after we published these papers, was when is the Warburg effect established? And that's what I will tell you in these next few minutes. Again, most of the people in the field of cancer metabolism believe that this was a late adaptation. Rapidly proliferating cells that are in need of doing Warburg for biomass, while they are acquiring more mutations and becoming more proliferative, they need to acquire this adaptation to proliferate. So it was considered that stage 3, stage 4 cancer were mostly doing Warburg. We have some data that suggested to the contrary, that this may be an adaptation that is established very early during the process of transformation. For instance, if you look at the G60 NCI dataset of RNA expression, you can see here the data for pancreatic cancer and colorectal cancer, and you compare a normal tissue to carcinomas, and you see a clear decrease in CYP6 levels accompanied by the increase in the glycolytic genes. In colorectal cancer, we can look at stages. And we saw nicely that even at stage 1 colorectal cancer and in adenomas even, CYP6 levels was already low, glycolytic genes were already high. This was not a late adaptation of rapidly proliferating cells. So we decided to ask in colorectal cancer when during the process of transformation this glycolytic switch happened. And my postdoc at that time, Carlos Sebastián, he's now a professor in Barcelona, asked, so we know that stem cells, normal stem cells are highly glycolytic. So he said, Raul, what about the tumor initiating stem cells? maybe they're already carrying some of this metabolic adaptation. And actually, the intestine is a very unique system to look at this. You're probably familiar with the original description of Hans Kleber of the intestinal stem cells sitting in the crypt of the intestine. These are LGR5 positive cells. What you may not know is that if you eliminate the APC tumor suppressor from these LGR5 cells, you get full-blown adenocarcinoma. If you do that from the transitional amplifying cells, you get nothing. So Hans Kleber defined the tumor-initiating stem cells in the intestine as cells that are derived from the normal intestinal stem cells. So we have a perfect system. We said, let's look on whether the cancer-initiating stem cells are already glycolytic. To do that, we needed to first define as a control metabolism in the normal intestinal stem cells. And so we look at our conditional CIR6 animals, and to our surprise, already in the normal cells, these are the crypts of the animals, and the CIR6 deficient in the intestine were increasing the normal intestinal stem cells in these crypts. Higher number when you don't have CIR6, and if we inhibit glycolysis by giving dichloroacetate, we saw a major rescue of this increase, suggesting that pushing glycolysis was increasing the number of these normal intestinal stem cells. We were surprised. We decided to test this in organoids, and we saw the same phenotype. For this, we generated a reporter where we put the promoter of PDK1, one of the glycolytic genes I mentioned, followed by the M-cherry reporter, and we saw beautifully rare cells in the intestinal organoids lighting up with the reporter. And we confirmed this by using a staining with an antibody against the phosphorylated form of PDH, the substrate of PDK1. And we got exactly the same staining. So rare cells are lighting up with this glycolytic reporter. So we were sure that this will be the intestinal stem cells, LGR5 positive cells. Now, these cells, as opposed to other somatic stem cells, are rapidly proliferating. And Hans Kleber showed it very nicely. So we thought maybe that's a reason why these normal stem cells are doing Warburg. And this came as a surprise when we stained with both a PDK1 cherry reporter and a marker of proliferation. We saw mutually exclusive staining in the organoids here, where the red cells that are glycolytic are not dividing, and vice versa, suggesting that whatever is this population that is glycolytic is not the LGR5 dividing cells. We were very surprised by this result, and we said maybe this is a weird phenomenon in the normal cells. Let's look at the colon cancer. So we took an APC transformed organoids, we put the reporter infected with the PDK cherry, we injected into mice, and we look at tumors. And we got in the tumors exactly the same pattern. First of all, extremely heterogeneous staining for the PDK cherry, and those cells that were doing glycolysis were not dividing in the tumor. So at least in the context of colon cancer, Warburg was not happening in the rapidly dividing cells. So what were the cells that were glycolytic in the normal intestine? So we went back to the literature. There is a population called the +4 stem cells. These are quiescent, differentiated enteroendocrine cells. They are fully differentiated, but if you eliminate the LGR5 cells, then these cells have the capability to de-differentiate, become stem cells, and populate the intestine. So we asked, since they were quiescent, maybe these were the cells that were highly glycolytic. And indeed, this was the case. We stained with a marker from these enteroendocrine cells, chromogramin A, and we saw perfect co-staining with the phospho-PDH antibody. And lastly, we were asked by reviewers, well, you didn't prove that these highly glycolytic cells, if they are quiescent, are they driving tumorigenesis? So we sorted the cells out of the mice, and we divided the sorting by M-cherry high and low. All the M-cherry low cells, even up to 1 million cells injected, never gave us tumors. And if we injected M-cherry high positive cells, as low as 10,000 cells were already enough to drive tumorigenesis in these animals. So why quiescent cells are doing Warburg? We follow this, not, I would say, one result that suggests why these cells are doing Warburg is actually to reduce reactive oxygen species. You have here one of these phospho-PDH cells with a marker for reactive oxygen species surrounded by neighboring cells. The neighboring cells show more ROS, and we quantify this, suggesting that the reason some of these quiescent cells, both in the normal and in the context of tumors, are doing Warburg is to protect them from reactive oxygen species. So this is what we show in the intestinal model, we believe that these +4 quiescent cells are glycolytic to reduce mitochondrial respiration, reduce ROS production, and since they are quiescent, these protect their genome that is sitting there for a long time. The problem is that one of these cells acquire a mutation, for instance, in APC, they may be coming with a head start, metabolically speaking, and that's why we believe we see more tumors derived from the CIR6 deficient animals that are highly glycolytic. And the one message I want to give you from this part is the highly heterogeneous feature of metabolism in this case. Most of the people studying cancer metabolism, they take chunks of cancer and they use this chunk of cancer to do metabolomics and determine how metabolism look in cancer. You cannot do that if metabolism is an heterogeneous feature of cancer. So we need to come up with creative ways to study metabolism if metabolism is an heterogeneous feature in cancer. And I will come back in 2 slides to that. So in parallel, a year ago, we published another paper in squamous cell carcinoma with basically the same results. The only thing I will mention, so you can see here, GLUT1 as a marker of the glycolytic cells is only expressed in the basal cells of the squamous cell carcinoma cells. The difference from colon is here the cells are rapidly dividing. So here glycolysis or Warburg is required for biomass as well. And the CIR6 knockout cells have more of these tumors, bigger tumors, and the unique cells that are affected by glycolysis are these tumor propagating cells with a 4-fold, almost 3-fold increase in these tumor propagating cells in the knockout animals. Interesting, this heterogeneous feature is not only from the glycolytic side. You can see here the marker for histone K56 acetylation, which is a target of CIR6. We see an increase of K56 specifically in the basal cells of the tumor, again pointing to an heterogeneous feature of tumor metabolism. We did metabolomics, we confirmed by RNA-seq metabolomics in vitro, and we confirmed by RNA-seq in the cells the clear signature of stemness, glycolysis, and increase in pentose phosphate to protect these cells from reactive oxygen species. Now, reviewers for this paper did ask us to confirm that these very rare stem cells were doing glycolysis. Again, you cannot do mass spec for metabolomics, but we were lucky to collaborate with Natalie Agar from the Brigham and Women's Hospital. She's one of the few people in the world that can do what is called MALDI-MSI, mass spectrometric imaging, which basically you acquire, you quantify per pixel metabolite concentration. It's still low resolution, but we managed to do that in our tumors. And you can see here in one slide, you can stain with your marker for the tumor propagating cells. In the neighboring slide, you do metabolomics with this model. And we were able to show that actually reduced glutathione was clearly increasing the tumor propagating cells compared to the non-tumor propagating cells. So we managed to perform almost a single cell resolution mass spectrometry imaging, which I think is where the future of the field will go. So I have a few minutes for the second story. And I did mention briefly that we generated this pancreatic cancer model where when we deleted CIR6, the animal developed this very aggressive metastatic disease. And this is with only the KRAS alone oncogene mutated. Most of the KRAS alone never give you tumors. If we take out CIR6, highly aggressive disease, 50% of these animals with metastasis. And the same is true even in the context of KRAS P53. We are able to push the disease to the left, much faster development of tumors. And again, 50% of these animals with metastatic disease. At the time we were publishing this paper, a couple of stories came out from one of the first analyses comparing the genome of primary and metastatic disease in pancreatic cancer. And in both papers that came back to back show very nicely that if you compare the primary tumors to the metastatic disease, there was very little increase in gene drivers, additional gene drivers compared to what you already see in the primary tumors. And actually the paper from Andrew Feinberg went one step further to say, not only we don't see much changes genetically, the main changes, non-genetic changes that we see are epigenome reprogramming of metabolic genes. So this was right out our alley. And when we saw these studies that were correlative in humans, we said we can use our mouse model to try to understand what are these epigenetic changes that happen in metastasis compared to the primary tumors. And we decided to go with two different models to expand our horizons. We did both in pancreatic cancer and in breast cancer in a model of 41 cells that you inject into the mice, and in weeks you develop lung metastasis. And because the animals develop metastasis so fast, we hypothesized that they will not have time to acquire new genetic mutations, but rather they should be adapting to grow in this new niche by acquiring non-genetic adaptations. So we did RNA-seq from all these samples, compared the RNA-seq, and we got what we call a metastatic signature. And this was 136 genes that were changing up and down, commonly to all the different metastatic lesions. And long story short, and up to here, many labs have been doing this. We follow with one step further, which was we took the 94 upregulated genes from this group, and we did a functional soft agar assay, high throughput assay in soft agar, where each well we eliminated one of these 94 genes by using an shRNA virus. And in each one of these wells, we tested which of these genes that were upregulated, when we inhibit them, they cause changes in growth of these soft agar colonies. And by using this method, we identified a few genes, a dozen, that gave us clear inhibition of growth in soft agar, while at the same time did not affect expression of 2D growth or the growth of the primary cancer cells. And I will just tell you briefly and rapidly, so we leave some time for questions. We follow one of the genes here called GSTT1 that you can see here in the list of the most commonly affected genes and functioning in our shRNA screening. This is a glutathione transferase, and a very rare one. It's only expressed in the liver, not expressed in the rest of the tissues. And it works different from other enzymes in the glutathione pathway that is used to produce reduced glutathione. GSTT1 is one of the transferases that conjugate glutathione into metabolite and proteins. Okay, it's a conjugate enzyme. So you modify proteins and metabolites by putting a glutathione modifier on those, and it's used in the liver to detoxify xenobiotis. Very little known outside of the liver, no roles in cancer or metastasis for this gene. And we'll just rapidly show you some of our functional data. This is from the mouse PDAC samples and from human rapid autopsy program samples. You see basically in green, no expression of GSTT1 in the primary samples, massive upregulation in the metastasis. Here is by Western. These are few samples from patients from the rapid autopsy program here at Mass General Hospital, and you see massive upregulation of GSTT1 in the metastatic cells. The validation for the high throughput, if we did 2D growth, we see no effect for downregulating GSTT1, but complete disappearance of the colonies. And the same is true in vivo. If we inject these cells in this pancreatic cancer, we created cell lines from the metastatic cells. We grow them by injecting into the mice. The primary tumors, no effect by knocking down GSTT1. The metastatic lesions completely disappear. This is a massive metastatic-specific phenotype. Furthermore, if we overexpress GSTT1 in these cells, we start seeing metastasis in ectopic areas. And you can see here tumors in the diaphragm, shoulder mass, skull mass, when you never see this in other models of PDAC. So overexpressing GSTT1 is enough to making metastasis in ectopic areas. I don't have time to go through these are some data which cells are. This is highly heterogeneous, same as I told you before. For the Warburg effect, we see this GSTT1 expressed in some rare cells. I will not have time to show you why, but we try to understand what is unique in these cells that are GSTT1 positive. We generated an endogenous M-cherry allele to sort these cells out. We did RNA-seq, and we saw clear increase in biological addition and cell addition molecules upregulated, decrease in cell division consistent with these cells being slowly cycling. And lastly, we try to understand what is GSTT1 regulating, and we perform a couple of mass elegant mass spectrometry elegant samples analysis, where we either IP for GST1 or glutathione enriched proteins. We identify a common set of proteins, and among them were several extracellular matrix proteins, including fibronectin-1, that were glutathione-related by GST1. And if we downregulated FN1, we saw disappearance of these metastatic lesions in the lung from the GST1 overexpressing samples, suggesting that indeed the way GST1 is driving metastasis is by modulating modification of fibronectin-1. So this is where we are with this story. It was finishing, the paper is submitted, and we will see how it goes, but we believe we have identified unique modulators of metastatic disease that are upregulated in an epigenetic manner, and we believe this gives these cells an ability to grow in this new foreign niche. And with this, I will stop. I think I am right on time. These are all the people in my laboratory. I think I have a nice picture that are currently in my lab. And this was mostly the work of Cristina Ferrer that already moved on. She's a professor now in faculty in the University of Maryland, and Carlos Sebastian, who is now in the University of Barcelona. And these are the current people in the lab that we didn't have time to tell you about their amazing work. But with this, I will stop and take questions.
Hi, Raul. Yeah, can you hear me?
Perfect. Yes.
Hi, Raul. So you nicely show the phenotype of deleting SIRT6 and what it does to cancer, and then it becomes more metastatic and more aggressive. So I wonder, what about the converse? Say, can you specifically activate or transgenic express more SIRT6, and what happens to the tumor?
Yeah, so depending who you ask, Chaim Cohen did this work. He created the transgenic SIRT6 animals, and he published a big paper in Nature about extension of lifespan by overexpressing SIRT6. I had a long argument with Chaim to try to see how much of that phenotype was actually protecting these mice from tumors. And we are still arguing about it. He tells me that he believed he see less tumors, but not enough to have statistical significance. And he believed that the reason these animals are living longer are not related to cancer. I believe that a lot of the effect of this overexpressing SIRT6 had to do with the protection of against developing of cancer. But probably you need to analyze much more animals for confirming that phenotype.
So a related question is, do you think activation of sirtuin is a general protective mechanism against cancer?
General sirtuin is activating, definitely SIRT6 I think will help. Activating SIRT6 probably will help. I don't know about other. Many of these sirtuins are showing, hundreds of papers showing tumor suppressive effect for several of these sirtuins. So probably activating other sirtuins may also have a role, but I don't work on the others, so I can comment only on SIRT6.
Thank you.
Great. And while we're on that topic, there's a question in the Q&A about SIRT6 from Larissa Traxler. How is the binding of SIRT6 on the chromatin regulated?
That's an excellent question, and I can tell you that at least in normal cells, we haven't explored this in the cancer context. In normal cells, in the 2010 paper we published, we believe it's brought down as many other repressors by the transcriptional factor itself. We show that in the in-HIF targets genes, SIRT6 binds HIF1 alpha and is recruited by HIF1 alpha. If we knock down HIF1 alpha, we see SIRT6 going out. In the MYC targets, I didn't have time to talk about MYC co-repression by SIRT6. We see many MYC targets also co-repressed by SIRT6. If we eliminate MYC, SIRT6 is fly out. So there are many examples where the transcriptional co-repressor is brought to chromatin by the transcriptional factor as a way to modulate expression. And we believe this is how SIRT6 is reaching its chromatin targets. We haven't looked in the context of the cancer cell for it, but that's a good question.
And there's a second question that's came right after that that's related from Alexei Anufriv. Excellent talk. And it's a general question. Are there cancers driven exclusively by epigenetic changes?
Thank you, Alexei. You mean, the question is referring to metastatic disease, whether metastatic disease is, because we know cancers in general, of course, are driven by genetic changes and associated epigenetic change. I don't want to say that only epigenetic changes drives metastatic disease. And we know that in many cases of people has done inducible deletion of oncogenes, you eliminate some of these metastatic lesions. So I think the original drivers that drove the primary cancers are required to sustain metastatic disease. But at least from what is coming from genomic analysis in many new analysis of primary and metastatic disease, people don't see other genetic drivers coming up. So I'm tempted to say that it's mostly epigenetic changes that are driving the ability of these cells to grow in the new niches.
Great. Thank you. So I have a technical question, actually. I was really interested. So the MALDI MSI
Yes.
platform is amazing. So what is the, so you have beautiful data. What is the capacity of this? So can you look at multiple metabolites? Is it targeted? Can you just get a small number, or can you give us a feel for it a little more?
So at this point, it's still, I would definitely refer you to talk to Natalie more about it. It's still at its infancy, I would say. The data that I showed was, we were lucky to get enough quantitative difference for reduced glutathione. Many metabolites don't show up. We try to look at all the glycolytic genes and TCA cycle genes, and many of these we did not get enough resolution to look at them. The field is moving very fast. Also, when you look by pixel, it's not really cellular resolution. It's 20-25 microns right now. The new equipment that the Bruker is now developing, they will be even subcellular resolution for much more metabolites. So I think the field is moving there. So definitely it's worth keeping an eye because we will be able probably by using MALDI-MSI to get subcellular resolution for many other metabolites. Still is targeted, but we are moving the right direction.
That's great. It looks amazing. Peter, do you want to jump in? Peter Adams?
Yeah. Hi, Raul. Fantastic talk. That's very exciting. So this function for GSTT1.
Yes.
So can you tell us a bit more? What do you think the glutathionation of fibronectin is doing? And do you think it has any control in? Does it have a redox control, which is also implicated in tumor
no, we
genesis,
don't.
or do you think?
It's a good question. We don't believe it's redox. These enzymes don't participate in redox control. And the only function for GSTT1 is in the liver, and it's only on detoxifying molecules by adding glutathione as a conjugate moiety. They don't function in redox. Although it's obvious to think about it because of all the work from Sean Morrison on redox control in metastatic cells. We don't believe this is the case for GSTT1. We don't see fibronectin as the only molecule being glutathioneated. We follow that because we need to choose one or a couple.
Right.
Follow. We see several extracellular matrix proteins being glutathioneated. So what we believe is going on from these cancer cells, GSTT1 is glutathioneating several of these extracellular matrix proteins that are secreted from the cancer cells. This is unusual because most of the people believe that the extracellular matrix is controlled by the fibroblasts, the cancer-associated fibroblasts or CAFs. In our model, we see clearly secretion of these proteins from the cancer cells, which for us was surprising. First, and second, we believe these are mostly non-dividing cells that we see expressing GSTT1. So we believe the cells are sitting there, secreting these proteins to arrange the extracellular matrix in a way that support these disseminated tumor cells to establish in the new niche. Once they are there, and this is a question I got from Bob Weinberg when I presented this in a meeting that he was there, I said, Raul, the most important question there is not how these DTCs, the disseminated cells are there, how do they awaken? How do they start proliferating, which is the question in the field? And I say, Bob, I wish I knew how they awaken. We still don't know what makes these cells starting proliferating, but at least we believe we define a new mechanism for what allows these disseminated tumor cells to reach the niche.
So does a tumor cell matrix tend to have a higher level of glutathionation than a normal matrix?
That's what we see in our paper. It was not studied before. No one looked at modification of fibronectin. We started to look, and we do see increasing glutathioneated fibronectin 1 in our model. So we believe that may be one thing that is making those ECM proteins better into generating the ECM.
Okay, that's really interesting. Thanks.
Great, and Vera Gorbanova, you have a question?
Yes. Great talk, Raul. Thank you. Well, I thought I'll ask you a question because I get this question many times. So I want to see what you would answer to that when I talk about SIRT6 and its tumor suppressive role. That people would often ask me that, well, in particular types of cancer, actually SIRT6 is overexpressed. So, like, can you clarify it for me? What is it actually doing?
I thought you were going to ask me about the DNA repair phenotype, how much it contributes, but before the overexpression, not many cancers. The only one, if you look at the TCGA data, there is one type of tumor that overexpresses SIRT6 as opposed to see deletion. In most of the tumors, we see downregulation of SIRT6, but there is one exception, which is hemato-oncology, among malignancy, leukemias and lymphomas are clearly overexpressing SIRT6. And indeed, there is one paper by Ken Anderson and Dana Farber that show what he believes is the reason. We don't see changes in lymphoid cells for metabolism if you modulate SIRT6, at least not in our hands. What we do see is clearly changes in genomic instability from the DNA repair functions of SIRT6. And we believe that those tumors benefit from upregulating SIRT6 because they become protected against chemotherapy. And Ken Anderson did a very nice experiment where he deleted SIRT6 in those tumors. He saw a major increase in sensitivity against the chemotherapeutic agents. So that could be one reason on tumors that may select to overexpress SIRT6 to protect these tumors against genomic instability. In most of the other tumors that we look, it's deleted, and we believe it relates to the ability to change metabolism. And to the second part that I know you didn't ask, but I do get this question on DNA damage and repair. I don't think it's only metabolism. It's clear that SIRT6 is a key modulator of DNA damage. You show it, we show it, and I believe a lot of these tumors do accumulate genomic instability by not having SIRT6. But that just so it's an additional function.
All right. Thanks so much, Raul. That was a great presentation, great discussion afterwards. So
Thank you.
To move on to the next talk. So we have Zhixin. So next up is Zhixin Do, who's from Mass General and Harvard, and he's going to tell us about nuclear autophagy in senescence and aging. All right, looks good.
Let me unmute myself.
Now you're out of the slideshow mode. I don't know if that was intentional.
Okay,
there you go.
Great. Can you hear me all right? Okay, great.
Yeah.
All right, thank you everyone for coming here. And I too want to thank the organizers for putting everything together. So I go by Do. Do is my last name, but apparently it's much easier to pronounce than my first name, Zhixin, so I just go by Do. It's much easier. So I will talk about nuclear autophagy in senescence and aging. So I'll start by talking about cellular senescence, which is an important hallmark of aging. And then I will also talk about other hallmarks related to senescence, for example, how it contributes to genomic instability, epigenetics, and the loss of proteostasis. So I'll first talk about the cellular senescence. And we all know that cellular senescence is a stable form of cell cycle arrest. Right? So this is permanent shutting down of the cell cycle. And then from the normal to senescence, a lot of things happen in the cells. For example, you see the cells getting bigger, and then they are more enlarged and flattened, and then the nuclei also look different. So this is a very stable form of cell cycle arrest in comparison to the quiescence, in which the cells can still re-enter the cell cycle. But senescence, by definition, is a permanent loss of cell cycle. Now, a lot of things can induce the normal to senescence transition. For example, the cells can experience telomere attrition, as was originally discovered by Hayflick. In addition to the replicative senescence, other acute stress can also induce the normal to senescence transition, for example, in response to DNA damage or activated oncogene expression. Now, this induction of senescence can be beneficial. For example, when the cells are experiencing activated oncogene, the cells can turn on this stable form of cell cycle arrest program, and this is a potent tumor suppressive mechanism to shut down the cell cycle. In addition, senescent cells secrete a lot of pro-inflammatory cytokine. This will recruit the immune cells, leading to immune-mediated clearance. So there's certainly a beneficial role of senescence. Now, on the other hand, in the context of aging, we know there are a lot of senescent cells that are accumulated in the tissue, and then the accumulation of senescent cells will block the cell renewal and the regenerative capacity of the tissue. And also, the senescent cells secrete a lot of inflammatory cytokine, the so-called SAFs, and then this will cause chronic inflammation that contributes and promotes most of the age-associated diseases. So that's why, given the important role of senescence in cancer aging, there's a lot of interest to target the senescent cells, either by killing them, the so-called senolytics, or using drugs to block the inflammatory phenotype, the so-called the xenomorphics. Now, as I mentioned to you, so a lot of the things are happening in the senescent cells from the nucleus. So this is what a normal mammalian nucleus look like. You have the nuclear pore complex, and you have the double membrane nuclear periphery, and then there's this nuclear lamina meshwork beneath the nuclear envelope. And so these are the type 3 cytoskeleton. They provide the nucleus with mechanical strength, and then they also regulate the higher chromatin organization. And so we know a lot of the chromatin is anchored to the nuclear periphery through the nuclear lamina, and these regions are repressed for their gene expression. So a lot of things happened during this process in senescence. For example, one of the most prominent changes is the loss of nuclear lamina, specifically the lamin B1 isoform, and this will lead to the chromatin deassociated from the nuclear periphery, and then a lot of the chromatin organized into a heterochromatin foci. And then there are other changes for the nuclear envelope as well as the nuclear pore complex. So as I mentioned, one key hallmark of senescence is the specific loss of the lamin B1 isoform. And we have shown that the loss of lamin B1 is happening at both transcription and the protein level. And the protein level is mediated by degradation by autophagy. So this is an example from primary human fibroblast. And you show in the control cells that lamin B1 is nicely localized at the nuclear periphery. And when you induce senescence by multiple means, what you see is that the overall intensity of lamin B1 is reduced, and you also start to see the cytosolic structure that is also positive for DAPI and for specific heterochromatin marks. And so Peter Adams' lab originally discovered the cytosolic chromatin structures, and then we collaborated with his lab by showing that the cytosolic structure can actually be targeted by autophagy for degradation. So in the next slide, I'll briefly introduce what is autophagy and what is this molecule LC3. So autophagy is a catabolic membrane trafficking process, or called self-eating, that's initiated with the formation of isolation membrane, and that later on forms the double membrane autophagosome, and that later on fuse with lysosome, and the content is degraded by autolysosome. Now, this molecule LC3 is a very important molecule involved in the autophagy process. So LC3 in the normal condition is a diffuse pattern, but in the setting of autophagy, so it's recruited to both outer and inner membrane of the autophagosome membrane. So the inner membrane pool of LC3 will recruit the substrate, so it directly binds or indirectly binds to the substrate and send the substrate to the autophagosome for degradation. Now, upstream of LC3, there are a lot of autophagy genes regulating this process, for example, ATG5, ATG7, and among others, and then they respond to starvation and specific cellular stress. Now, this degradation process can be fairly non-specific. For example, in the context of starvation, the cells are trying to grab whatever they can eat to get through the hardship. So this is considered bulk autophagy or non-selective. But on the contrary, autophagy can be fairly specific. For example, in the process with senescence, the cells use autophagy to specifically degrade specific substrate for a particular function. So what is really weird about this autophagy adapter molecule LC3 is that in the basal state, in the control sense, right? So you see a lot of LC3 is in the nucleus, and then in the starvation or stress condition, the nuclear pool becomes empty, and then the cells form this autophagic puncta that has LC3 on it. So this has been very confusing to the field. what is LC3 doing in the nucleus. And so we were one of the first to show that there was a nuclear role for autophagy, and especially for the LC3. So what happened is that part of the LC3 in the basal state is in the nucleus, as I show you, and then a portion of the LC3 directly binds to the lamine B1 isoform and chip at the lamine-associated domains at the heterochromatin. So this is what happened in the basal state. Now, in the process of senescence and aging, what happened is that the nuclear membrane forms the bleb, and then these structures can also butt off to the cytosol to fall off to become cytoplasmic chromatin fragment that has LC3 with lamine B1 and part of the chromatin. So they can undergo autophagy degradation, and then we have shown that together with Peter Abrams' lab, and this is a major mechanism to lose the lamine B1 in senescent cells as the protein level degradation. So in addition, the cytosolic DNA is sensed by the innate immunity cytosolic DNA sensing pathway, CGAS-STING pathway, that contributes to the senescence-associated phenotype. So we do think the autophagy, especially the nuclear autophagy, has a very important role in senescent cells in altering the nuclear and chromatin structure, leading to gene expression changes. So these studies are all published. So for the rest of my talk, I'm going to talk about some unpublished work from my lab, asking the question, does autophagy degrade any other nuclear substrate in senescence? Right, so I've shown you the lamine B1 as an example. Usually, if you see one, there's got to be more. So we asked a very broad question, are there any other novel substrate of nuclear autophagy in senescence? So we collaborate with Hopper's lab in Harvard to take the unbiased proteomic approach to find novel substrate of autophagy in the nucleus from the senescent cells. So our approach is that we first ask what is the nuclear LC3 interactome, what binds to LC3 in the nucleus, and then we also ask so what is specifically lost in the senescent nuclei, and we're comparing the autophagy competent versus autophagy deficient nuclei. So the overlapping pool is what we are most excited about, right? So that has the highest confidence of the putative novel substrate of autophagy. So what did we find? So the top hit is lamine B1, which is good. That serves as a positive control. So the other target we found is a weird protein called WSTF. So this protein is never implicated in autophagy or senescence. So I'm going to tell you what it does and what is the biological function for WSTF in senescence. Okay, so WSTF is in fact part of the I-SWI chromatin remodeling complex. Right? So we know that normally the DNA is organized into nucleosome, which repress the gene expression and transcription. And that there are chromatin remodeling complex that can open the nucleosome to expose the DNA, so activating the transcription or replication. And they are also complex that doing the opposite, that can switch the open chromatin back to closed chromatin. Now, I-SWI complex is a repressive chromatin remodeling complex. So it uses ATP and shift the open chromatin back to the closed chromatin, thereby repressing gene expression. Right? So I-SWI complex is a repressive complex to repress gene expression. So the I-SWI complex in mammals has a lot of different subunits. The catalytic enzyme, the ATP is the SNF2H. And then this WSTF is a particular regulatory subunit that directly binds to SNF2H. So what is really interesting is that from our proteomics data, so you only see the specific loss of WSTF in the complex, and you do not see loss of SNF2H or other complex. So indicating the cells has a very clear idea what it's trying to do. So first thing first, let me show you some validation. And we have shown that the loss of WSTF at the protein level is a common signature for cellular senescence. For example, this is IMR90 from a human fibroblast. You see when the cells are senescent, they specifically lose lamine, the WSTF, and then the catalytic subunit, the SNF2H, remain largely unchanged. This is also true in mouse and cancer cells undergoing the therapy-induced senescence. And what is also interesting is that if you look at the mRNA level, WSTF is completely unchanged. So we know this is a very specific protein level degradation. Okay, so obvious question is autophagy really involved in WSTF loss in the senescent cell? So we are answering this question using both genetic and pharmacological approaches. So first, we stimulate knockdown the autophagy essential gene ATG7. As you see, the knockdown of ATG7 block the LC3 lipidation, so the cells are really autophagy deficient. And then what you see is, interestingly, in the control cells, when you induce senescence of the fibroblasts, you'll have a very nice drop of the WSTF at the protein level. And then when you don't have ATG7, the protein level largely stay there. So we can also block autophagy using drugs. So now we induce senescence, and then you see a very nice loss of WSTF. And on top of that, we can treat the cells with the lysosomal inhibitor buflomycin, and then you see the WSTF protein is largely reversed. Now, by contrast, if you treat the senescent cells with the proteasome inhibitor MG132, you see nothing happen, WSTF is not reversed. So we are very clear that this loss of WSTF is specifically driven by autophagy in senescent cells. Next, we are going to the mechanistic insight, how autophagy, and specifically nuclear autophagy, degrade the WSTF. So I mentioned to you about LC3, and in fact, LC3 is a family, is a member of the ATGA family proteins. So there are 6 members, and then they recognize the different substrate under different conditions. So the 6 isoforms give the cells the complexity and the diversity to recognize the substrate. So we have done purified in vitro experiment in which we in vitro translated WSTF and used bacteria to pull them. And then what you see is that the GABA-RAB isoform of the ATGA is the predominant isoform that potently binds to the WSTF. So we have further mapped the WSTF and the GABA-RAB interaction in great molecular details, and that we know the specific residues on both ends that mediate the interaction. So on the WSTF side, that this linker region, and specifically the leucine residue, is really important in mediating the GABA-RAB interaction. Right? So when we truncate this short linker or we mutate this leucine to alanine, we almost completely lose the interaction with GABA-RAB. So the obvious question is now, what is the behavior of this mutant in senescent cells? It is still being degraded by autophagy. Right? So now we stably express either the wild-type WSTF or the 2 mutant that doesn't bind to GABA-RAB. And then what you see is that in the control senescent cells, the WSTF is nicely reduced by autophagy degradation. And then the mutant that doesn't bind to GABA-RAB largely stay there, indicating the binding to the autophagy adapter is really important. And we also see that in the wild-type WSTF, you see the nucleus of cytosol trafficking followed by cytosolic autophagy, lysosome degradation. And these 2 mutants largely retain in the nucleus, so they have trafficking problems. Okay, so after all that, so what does WSTF do in senescent cells? Why do the cells really want to degrade specifically the WSTF subunit of the ISY complex? Is there any biological significance to this? Right, so now, to do this, we counteract with what the cells are trying to do. So if you want to degrade it, we force to overexpress it. And then we ask the question, what happened to the cells? All right, so you see in the control cells, the WSTF is nicely lost, and we use a lentiviral vector to stably express WSTF, and we ask what happened to the cell. You see the loss of laminin P1 or the induction of P16 as senescence markers are not really affected. But what is interesting is that in the control cells, you see a nice induction of L8, which is a marker for the senescence-associated secretory phenotype. And then when you put WSTF back, the L8 expression is largely repressed. So now we can also do this at the genome-wide level to look at the RNA-seq. And then what you see is that in the control senescent cells, mediated by irradiation, there's a very prominent induction of most of the SAS specific, the inflammatory genes. And then when you put WSTF back, so the inflammatory response of the senescent cells are largely repressed. And then we can also show at the genome-wide scale that the WSTF overexpression specifically affect the senescent, the SAS phenotype, the inflammatory response, and it doesn't affect anything else. So now this is overexpression of WSTF. So we can also do the reverse by knocking down WSTF. And this is what you see. We can stably knock down the WSTF. And then again, it doesn't really affect laminin P1, P16, a loss of phospho-RB. But then you look at the inflammatory genes expression L6 and L8. You see when you don't have WSTF, these genes are massively turned on at a much faster speed. And so overall indicating that WSTF is a repressor of the SAS program of senescent cells. Okay, so the question is, mechanistically, how does WSTF block the SAS program of senescence? So we have looked at all the existing mechanism of the SAS. For example, Judy Campise's lab have shown that you need a P38 MET kinase ATN, and as well as the mTOR pathway to regulate the SAS. We have also looked at the cytosolic DNA sensing cGAS-STING pathway and the P53 pathway, and turned out that WSTF doesn't affect anything of that. So the question is, how does WSTF block the SAS program? Now, our hypothesis is that WSTF blocks the chromatin accessibility as part of the ISY complex. So remember, I told you earlier that WSTF is part of the ISY chromatin complex that is repressive. Right? So now our model is that in the control cells, the chromatin is largely closed. Now, in the senescent cells, if you want to turn on the inflammatory genes, you need to open the chromatin, and this will allow the transcription factor NF-κB to kick in to do its job to stimulate the expression of the inflammatory genes. Now you see if the cells are open in this stage, if they see ISY complex, right? So the ISY complex is going to shift the open chromatin back to the closed chromatin to repress your gene expression. And that what happens is that we think that autophagy specifically degrades the WSTF and the ISY complex. So you will shift the equilibrium more to the open chromatin, leading to the stable and sustained expression of the inflammatory gene. So to test this hypothesis, we are running ATAC-seq to look at the chromatin accessibility of senescent cells. Okay, so I'm going to give you one example of the L8 or CXCL8 genes. You see in the control cells, the chromatin accessibility is low over the L8 genes. And then when you induce senescence of the cell, you see the massive opening of the chromatin measured by the ATAC-seq signal over the L8 genes. And then when you put WSTF, and this chromatin opening is largely blocked. Right? So we can again do this at a genome-wide level to show pretty much all the inflammatory genes are affected, and then also WSTF specifically block the chromatin accessibility of the inflammatory genes, and it doesn't block anything else. And so here, I'm going to skip some in vitro data before I talk about the in vivo data to quickly summarize what we think is happening at the mechanistic level. So we think normally WSTF, as part of the ISY chromatin remodeling complex, will block the chromatin opening. The other thing I didn't have time to show you is that the WSTF directly binds to P65 subunit of NF-κB and also block the acetylation, which is required for the activation of transcription of NF-κB target. So these two mechanisms serve as a potent inhibitor of inflammatory response. Now, in the context of the senescence, the GABA-RAB binding to WSTF is specifically enhanced and greatly elevated. So this will lead to the nucleus to cytosol trafficking of WSTF, leading to autophagic degradation. And so then you relieve the inhibition, and this is how you turn on the inflammatory process of senescence at the chromatin level. Now, in addition to senescence, we can also show WSTF is a very broad and potent inhibitor of NF-κB mediated signaling in the context of senescence cancer and infection. So we found this is a constitutive nuclear inhibitor of inflammation, and that's why nuclear autophagy wants to degrade this negative regulator to turn on the pro-inflammatory response of senescence. So the last question is whether this process is also important in vivo models of senescence, whether you also see loss of WSTF in vivo senescence, whether this is functionally important for inflammatory response. So we use a model to stably express the activity oncogene NRAS V12 in the liver hepatocyte. So we clone the NRAS into the Sleeping Beauty transposon system. And we use a technique called hydrodynamic injection. This is large volume, high speed injection through the tailwind. So this will lead to the specific expression of the oncogenic NRAS in the hepatocyte. So this will lead to oncogene-induced senescence in the liver hepatocyte, and that will cause the inflammatory response through the SAS, and then they recruit a lot of the immune cells, leading to immune-mediated clearance of the oncogenic hepatocyte. So this is a potent tumor suppressive mechanism mediated by senescence and mediated by the SASP. So this model is very robust. So if you set up the injection in day 0, you start to see the peak of the inflammatory response at day 6. And then 6 days later you start to see the clearance mediated by the immune system, immune-mediated clearance. And if you wait for additional 6 months, you start to see the liver phenotype. Now, in the normal setting, I should say that because the red cells are all cleared off by the immune system, so you don't really have to tune. But as we have, others have shown that if you block the inflammatory response, for example, by knocking out cGAS or STING, you start to see the massive accumulation of liver tumor because you lose the immunosurveillance of the oncogene. So what we are doing is we are either expressing GFP as a control or to force the cells to express the WSTF together with the NRAS. Okay, so first thing first, whether we can see WSTF loss in this model. So this is the day 6 of the injection, and then you start to see the NRAS expression in the liver. And then what you also see is that when you see the NRAS, you also see loss of WSTF in the nucleus. Right? So when you see NRAS, you also see the loss of WSTF in the nucleus. So normally the hepatocyte expresses very nicely amount of the WSTF, but when you express NRAS, they enter senescence, you also see the loss of WSTF. So this gives us the justification to put WSTF back. And now we can ask the question, what happens to senescence? What happened to the SASP? Right. Now, this is the day 6. As I mentioned, this is the peak of the inflammatory response. You see in the GFP group, you have the NRAS expression in the liver. And that we also stand for CD45 as an immune cell marker. So what you see is that there's a massive immune cell infiltrated to the liver. And then what they are trying to do is to clear the oncogenic cells. Right? So what happened is that the NRAS expressing hepatocyte enter the oncogene-induced senescence. So this secrete a lot of the inflammatory cytokine, the so-called the SASP. So this will recruit the immune cells and activate them, and then leading to immune-mediated clearance of the oncogenic cells. Now, what happened on the contrary is that if you force the liver to express WSTF together with NRAS, you still see that these NRAS are being expressed in the liver, but what you don't see is the CD45 infiltrating immune cell because there's no SASP, and then there's nothing to tell the immune system to come over, so then you don't really have the infiltration of CD45 positive immune cells. So this is day 6, peak of the inflammatory response. Now, starting in day 12, you will see the clearance of these cells by the immune system. All right, so you see in the control GFP group, the NRAS cells are largely gone because there's still this massive CD45 positive immune cells forming clusters. They are trying very hard to clear the oncogenic cells. And then what you see in the WSTF overexpression group is a total different scenario. You still have a lot of oncogenic cells, and then you don't have the CD45 positive immune cells coming over because, again, you lose the SASP. Okay, so what happened to this liver? They have tumor. Right, so you see in the control liver after 6 months, nobody have liver tumor because the immune system took care of it. Right, so this is a potent immune-mediated clearance model mediated by the SASP. Now, if you look at the WSTF overexpression group, as I showed you earlier, there's no immune cells being recruited because there's no SASP. 100% of these mice develop massive liver tumor because you lose the immunosurveillance of the oncogene. So again, showing you that the loss of WSTF is really important in mediating the SASP phenotype, and that in this particular model, and then this is also a potent tumor suppressive mechanism. So you force the cells to put back WSTF, and you lose the tumor suppression. Okay, so to summarize everything we talked about, so we think WSTF is a novel nuclear substrate of autophagy. So we have shown the lamin B1 is the first substrate of mammalian autophagy in the nucleus. So this is through an unbiased proteomics approach. We think we identify WSTF as a very important substrate of nuclear autophagy in senescence. And then functionally, loss of WSTF by nuclear autophagy is really important to turn on the SASP program. And then I've shown you, if you put it back, you block the senescence. You block the inflammatory phenotype, and that has a very detrimental role in terms of tumor suppression. And we also show that WSTF is a direct binding partner of NF-κB and block the NF-κB acetylation. And this is WSTF is a potent nuclear inhibitor of NF-κB mediated inflammation. So what we think autophagy, nuclear autophagy is doing is that we think this is intrinsically a cell stress response. So the cells will turn on the nuclear autophagy, coping with the cell stress, and this will degrade laminin B1 and WSTF together with other substrate that we are still characterizing. So this will lead to profound nuclear and chromatin changes. And this, depending on the nature of the substrate, for example, we have shown the loss of laminin B1 is very important in reinforcing the cell cycle arrest. And then degrading WSTF will trigger the inflammation. We think this is beneficial in that it's a potent tumor suppressive mechanism. But depending on context, if you look at the natural aging, this will also block the tissue regeneration and will also contribute to chronic inflammation. So this is consistent with this idea that something is good for the young can turn out to be bad for the old. So that's why we think could be a potential therapeutic targeting window that we block the autophagy degradation of the specific substrate to promote healthy aging. So this work is mostly done by Yuan, a postdoc member, a postdoc fellow in our lab, together with Angelique. And then we also acknowledge Wade Hopper, who contributed the proteomic work, and Torgeir Johansen's lab in Norway, who help us to characterize the autophagy mechanism. So the most important take-home message today I want to deliver is that we are recruiting postdoc fellows. So my colleague Jay Rajgopal and our lab is recruiting a postdoc fellow to work on senescence, aging, and infection, and the lung. So we are part of the NIH Senescence Network Symposium, the consortium, and we propose to use single-cell proteomic approach to study senescence and the lung. So if you are interested in epigenetic senescence, aging, and the lung, definitely consider us. Yeah, so I'm going to stop here, and I'm happy to take any questions you may have.
All right, that was fantastic. Really beautiful work.
Thank you.
Right. I see lots of questions coming in, so I'll go ahead and get started from the Q&A here. So from James Haslam, he wants to know, what's the method you use to find interactors of LC3 specifically in the nucleus?
Right, so this is a very good question. So what we did in this case, we specifically fractionate the nucleus, and then we performed the ATG8 IP mass spec from there. And then we are actually doing the 6 isoforms, and then they show overlapping yet distinctive binding partners. So short answer is we fractionate the nucleus and specifically looking at the nuclear interaction. I do think this is a lot of people have done the LC3 IP mass spec before, but they are looking at the whole cell or the cytosolic fraction. So we specifically fractionate the nucleus.
Great, thanks. Aaron Havas, did you want to unmute and ask your question?
Yeah, Do, wonderful talk. I'm always excited to see your work.
Thank you.
So you did hone in on the NF-κB effect of whenever you target WSTF, but I think early and late senescence, right? So are you specifically just targeting the NF-κB like arm, or do you think you're affecting interferon as well?
Right. So we have not looked at the interferon response yet in our model because we are characterizing an early time point of the senescence. I believe the interferon response is a much later process, as reported by John Sedley, also by Vera, so that you need to turn on the line 1, which I also believe it activates the cGAS-STING response. But we have not looked at the interferon response in our model.
But you can in your mouse model, right? You did take that out quite far. If you have earlier time points, you could easily look for like some of the IRS, yeah?
That's right. That's right. That's something we can certainly look at. For example, in the fibroblast model in vitro, we can wait for much longer to look at the interferon. And then we can also look at the mouse models, of course.
All right, great. So there's another question from the chat. This is about the relationship between senescence and I guess normal aging. So Kristen Geronimo, degradation of WSTF1 by nucleophagy is a mechanism associated with senescence, can it also occur in normal aging?
Right. So the question is whether the WCF, loss of WCF is a process occurred during natural aging. So we have looked at the aged mouse compared to the young mouse. We have looked at the tissue by tissue, and that we do not see loss of WCF in most of the tissues, right? So, but what we have seen is that WCF is specifically lost in the senescent cells. So when you have the senescent cells in the aged tissue, you start to see loss of WCF just in that senescent cell, but not in the broad tissue. So we are actually testing this idea. Loss of WCF could be a potential marker for senescence, just like loss of lamin B1. You don't really see that in most of the tissues, but you specifically see loss of lamin B1 in senescent cells. But in a way, lamin B1 is also degraded by nuclear autophagy. We think the lamin B1 and the WCF is probably shared in the same pathway.
Great. And while we're on the topic, there was another question about the LC3 and the lamin. Does LC3 also recognize progerin?
They are papers showing that in the progerin model that you start to see co-IP between LC3 and progeria. And I also know there are papers showing the rapamycin, when you activate autophagy, you facilitate the degradation of the progeria in the mouse models, and associated with the extended lifespan of the progeria model. So yeah, so short answer is yes. So this is not from our work, but from what I heard and read, I do see that LC3 can co-IP with progeria.
Great. So in terms of the chromatin accessibility work that you've been doing, is it known, this is an anonymous attendee, is it known what regulates the opening of the chromatin for SAS genes in the first place?
Right. We have not looked at that yet because we are looking at the repression. Right? But I believe a lot of other people have looked at that from the histone modification level. You have acetylation as well as the methylation that contributes to the chromatin opening. So we have not looked at that. This is from other works, other people's work. But I also think it's equally possible that there are chromatin remodeling complex, for example, the Swiss-SNF complex, that can open up the chromatin. But what we have shown here is the ISY complex can close the chromatin. And that's why autophagy wants to degrade it.
Great. And so Sweta Sikder had a question also related to the chromatin remodeling data. She found your talk very interesting, and she's curious to know how much of the ISY chromatin remodeling activity is affected upon loss of WSTF alone.
So the question is whether you really need senescence or you simply disrupt WSTF, you start to see the chromatin opening phenotype. So what we have shown is that in terms of activating the transcription of inflammatory genes, you absolutely need the transcription factor, for example, NF-κB in this case. And that NF-κB in the normal setting is floating around in the cytosol only in response to inflammatory or the infectious signal does NF-κB translocate to the nucleus. So what we know is that you need WSTF to work together with NF-κB in order to turn on the transcription factor, to turn on the transcription of inflammatory genes. You simply knock down the WSTF, you don't really trigger the inflammatory response, but you lower the threshold, and then the cells are more sensitive to inflammatory cytokines. So as I mentioned, we do know that WSTF directly binds to the P65 subunit of NF-κB. Right? So we do think they need to work together to turn on the transcription response. And that's why autophagy wants to get rid of WSTF to boost the inflammatory response.
Okay, great. And then there was a second part to this question. So in your ATAC-seq data in the senescent cells, do you find that chromatin accessibility is sustainably affected only at the SAS gene? So I guess, is there a broader response as well, or broader effect genomically?
Right. So that the question was to ask what is the process comparing proliferating versus senescence. Right. So what we have shown you and what I mentioned to you is the senescence versus senescence stably expressed WSTF. So when we look at what is globally open in the senescent cells, right? So you do see there's a massive upregulation, massive opening over the inflammatory genes. So we have that data, but I forgot right now whether you do see other changes but it wouldn't surprise me there was also a close of the cell cycle regions together with other gene regions. So we have uploaded the data to GEO, so everyone is more than overcome to look into that.
Yeah, that's great. I'm sure some people will follow up on that. It's a beautiful data set that you have. So one more, Carla Valdivieso, probably apologize, probably pronounced that wrong. It's about cause and effect. So do you think the loss of lamin B1 is triggering the activation of P16, or is it the other way around?
Right. So this is a complicated question regarding the relationship between lamin B1 cell cycle and P16. So what is already known is that, so first you do have loss of lamin B1 in senescence. Right? So when you put it back, you delay the senescence, you delay the cell cycle arrest, and then you also delay the P16, P21 response. So it's hard to say what happens first, what happens later. So in a way, this is kind of a chicken and egg question. So from our data set, we also know that when you put lamin B1 back, you delay the P16, P21 response. But as I said, this could be you block the cell cycle arrest. So that's why you block the P16, P21. So I see that the P16 is strongly regulated at the epigenetic level. So it's not going to surprise me that there's some crosstalk between lamin B1 and the P16. So one more thing I want to mention is that the loss of lamin B1 is really important for the senescence-associated heterochromatin foci, the STAPI foci in the center of the nucleus that are repressive. And so if you lose lamin B1, this is very important for the formation of the SAF heterochromatin foci. In the senescent cells, you can put the lamin B1 back, and then you see the heterochromatin foci will disappear. So the loss of lamin B1 is very important for the epigenetic reorganization in senescent cells. So it's very likely to directly or indirectly talk to P16.
All right, thank you so much. That was a fantastic talk.
Thank you.
And last up, we have Dr. Berenice Benayoun, who's at USC, Southern California, and her title is Sex-Specific Remodeling of Aging Murine Macrophages.
Thank you, Ashley, and really thank the organizers like Peter for putting together this event. I think this has been really exciting, and hopefully I can maintain excitement. So like Ashley said, today I'm going to tell you about some of the work my lab's been doing to understand sex-specific patterns of omic and functional remodeling with aging in the immune system, and here specifically macrophages. And I'm sure you all saw a couple weeks ago an updated Hallmarks of Aging came out, and I was actually really excited to see that it now includes chronic inflammation as one of the hallmarks, because this is really something that we and others have seen being really a recurrent feature across tissues, cell type, species as being really characteristic of aging. And so this has also been sort of highlighted and coined under the term inflammaging by Claudio Franceschi, this idea that with age, and probably due to repeated exposures during the lifespan, there is a chronic activation of the innate immune system leading to this chronic inflammatory state that might in and of itself be deleterious and create some of the phenotypes of aging. And so in my lab, we're really interested in understanding how that happens, but also with a highlight on how sex dimorphism and biological sex plays a role in this. And this is why I like to say that with the way people normally do aging research, we might be missing half of the picture. I think Ashley, Dr. Webb did a great job at the beginning of the session explaining some of the issues in the field with the overwhelming majority of studies that have been conducted in the aging field, especially when there's a molecular angle to it, being done exclusively on male animals. And when you talk to people, there's usually a lot of explanations as to why they chose to do only males, ranging from that it's too expensive to do both, to female data is too noisy, to my favorite, females are just males with hormones, which by the way, they're not. So, you know, if there's one thing you get from my talk today, please let it be that. But because of that mindset, we're in a situation where we know very little about differences in the way females and males age, especially at the molecular level, which is where we can really get insights into potential therapeutics. And so why is that important? Well, it turns out that over the past decade or so, like maybe even more now, we've had a great NIH-supported program, the NIH Interventions Testing Program, to sort of cleanly determine what interventions are going to have an impact on, slowing down aging, or at least delaying the health issues linked to aging. And I think what the biggest finding from this consortium has not been that this drug or that drug can delay age-related decline, but really that there's literally not a single intervention that they've tested that works the same in females and males. You have drugs that work exclusively in one sex, or drugs that work much better in one sex than the other, which really suggests, at least to me, that if aging was exactly the same thing in females and males, then you would expect the same drugs to have the same effect in both. And the fact that they don't means there are some fundamental differences that we need to better understand, so we can design longevity interventions that are tailored to individuals. And there's a lot of other things telling us this is not just going to be a mouse specific issue. We know that menopause in humans is literally the single biggest risk factor for women to develop all sorts of age-related diseases, including Alzheimer's, for instance. Suggesting that reproductive function, reproductive hormones, and sex in general will impact the way you age. A big consortium like the NIH Roadmap Epigenomics Consortium showed, and they were really surprised by this, that the biggest difference in every single tissue where you do epigenomic profiling, the biggest difference is not the age of the individual, it's not the disease status of the individual, but really whether the tissues come from female or male individuals. And so I told you we're really interested in the immune system. Well, it turns out there's a lot of reasons to believe the immune system itself, regardless of age, is very sex dimorphic. We know, for instance, that for every man that has an autoimmune condition, you can find 9 women. So suggesting potentially a more active immune system, at least in young women. But conversely, we know that outcomes after infections or sepsis are always much worse in men. So again, the immune system, clearly sex dimorphic. How is that influenced with aging? And so this is a question that a very talented graduate student in the lab from the Biology of Aging program, Ryan Liu, has been interested in elucidating. And he started really with the question of whether we could identify and understand sex dimorphism in the aging immune system. And so at the time, my lab was very new in the immune domain, and so we wanted something sort of easy to access that we could study. And it turns out the peritoneal cavity, where most of your organs are sitting, is sort of ticking all the boxes for this kind of study, because it has a well-defined tractable immune ecosystem that has various immune cells belonging to both the innate and adaptive compartments like macrophages, B cells, T cells. It's also important in the general homeostasis of the cavity, but also in response to acute issues like peritonitis, so it has physiological relevance. And then finally, it's also very easy to access through postmortem peritoneal lavage. You don't need to do any tissue dissociation that can perturb cells and their gene expression and their function. You can just get the cells using a PBS-BSA injection in the peritoneal cavity postmortem. And so all of this gave us like a really nice way to study what's going on with age in an immune niche. And so we used aging mice. And so we have, in this case, in each experiment, only one provider, but we have both mice from the aging mouse colony at NIA and some aging JAX mice, and we see very similar results in both. So I won't specify which one came from which moving forward, but we get very similar results from both vendors. And so we use, for the young time point, we use 4 to 5 months of age. We do this because this is about when the hormonal cycles in young female mice are settled. So instead of looking at teenagers, we're actually looking at young adults, at least in that angle. And we compare them to old, 18 to 20 months of age. And we use that age frame because at least in the C57 Black 6 background, by 22, 24 months of age, about 80 to 90% of the mice have pituitary adenoma, which we're afraid might give us a false idea of what might be happening in hormonal axes. And so, as I said, we perform peritoneal lavage, and then sort of to unbiasedly figure out what's going on in that niche, we used single-cell RNA-seq using the 10x genomics platform. And so the good news is that we are able to find the cells we're supposed to find in that niche. We have a majority of B cells, and then we have a large population of macrophages, some monocytes, and a good number of T cells as well. And so not only we can identify what the cells are, but we can actually see which of those cell types, once we annotate them, are most responsive to the biological condition we're studying. So in this case, being young or old, female or male. And using a framework called OGRE, we can determine which of those cell type is most responsive transcriptionally. And what we found, and you can see here because that's the most intense red color, is that the macrophages in this case are the most responsive to female versus male, young versus old conditions, suggesting that they would be the most interesting for us to study. Now, we can also look at the relative abundance of those cells in the different libraries and see if there are changes in the composition. And we notice, and this has been observed by other people in either females or males, but never together, that we have a huge shift in the proportion of the cells in that niche, with an increase in the B cell fraction and a decrease in the macrophage fraction. And it looks like those changes are a little bit more extreme in the females than the males. For the T cells, we don't think this is real biologically because we've done flow cytometry and we were able to confirm B cells and macrophages, but not the T cells. And so at this point, the macrophages, we know they're changing in proportion, and we know they're changing at least at some level in their gene regulation. Now, single cell is very good for a lot of studies, but the problem is it suffers from a lack of sensitivity on the lower range of gene expression, just due to the way the technology works. And so since macrophages are very well-defined cell type, we could actually go back, purify them, and characterize them using bulk methods that are way more sensitive in that manner. And so to do this, we use the same type of mice. We do the peritoneal lavage. We purify the cells to get very high purity of F480 positive, CD11B positive, which those are those mature macrophages in the peritoneum, and then perform a multi-omic characterization of the cells for the transcriptome, and then their chromatin and epigenetic landscapes using ATAC-seq and cut and run for H3K4 trimethylation. And so what do we see? Well, what's really interesting, and so since those are very high dimensional data sets, we usually like to first project those in a 2D space to see what are the big differences, right, between the different samples. So this is multidimensional scaling. So what we observe, which is consistent with what I told you during the sort of the introduction of my talk, is that the biggest separation here in the transcriptome is actually whether the cells were purified from males or female animals. And there is a secondary sort of separation when you look between the darker colored and the lighter colored dots, suggesting there is a secondary axis for aging, but it's a little bit mixed. So it's definitely not as clear. Sex is definitely the biggest change in the transcriptome, and we observe genes that are significant with both variables. When we perform the ATAC-seq analysis, we see a very similar picture. If anything, the difference between the female and the male's macrophage is even bigger. And same for the cut and run for H3K4 trimethylation, very clear separation between female and male samples, and secondary separation for aging. And so here it is very clear that at the basal state in young, healthy animals, those cells are different. But in our lab, since we're interested in how, the difference of the trajectory, right, how they age, this does not tell you if there's a difference in trajectory. It just tells you there's a basal difference you're working with. And so in a very sort of naive way that we look at this is we say, okay, well, even if they start from different spaces, we can look at how they react to aging by looking at the rate of change in gene expression with age in the female macrophages here on the x-axis, compared to the rate of change in gene expression with age in the male macrophages here on the y-axis. And the idea is that if they go along very similar trajectories with aging, this should be very correlated. And so although there is a significant correlation, this is a Spearman rank correlation of 0.288, this isn't super impressive, I'm sure, to any of you. And so that suggests that there is not a great correlation. And so if we move a little bit further and we look for genes that were significantly changed with age only in the females or significantly changed with age only in the males, those are labeled in pink and blue dots. And you can see that there is definitely a nice population of genes we can identify, and no clear pattern of where they are in the graph, suggesting that they're mostly decorrelated with each other and what's going on with age on those genes, suggesting that the aging trajectory of those peritoneal macrophages are largely sex-specific. So, it's great. We have genes changing. We have peaks changing because we have a similar view when we look at the ataxic and the cut and run. But really, we're biologists. We're curious about what that means biologically. And so we decided to perform enrichment for gene ontology terms in the female and the male macrophages with age, and then focused on the pathways or the terms that end up being significant only in one sex with age. And this here, I'm showing you only the top 10 in each sex. So here in that first column is the top 10 most significantly regulated pathways in female aging macrophages, and in blue, the top 10 pathways most significantly regulated in male aging macrophages. And we noticed a couple of things. Notably, here there's phagocytosis recognition pathway and some membrane pathways here, the endoplasmic reticulum, plasma membrane, suggesting that maybe something's going to be going on with phagocytosis, which is a key function of those cells. Now, the problem is gene ontology in general has a very poor annotation for phagocytosis regulators. So we also wanted to confirm this using better lists. And so for that, we took advantage of 2 CRISPR screens that were published in the past few years that identified over 500 new phagocytosis regulators in macrophages and macroglia. And using those and performing GSEA like we did in the previous analysis, just for those phagocytosis regulators, we see a very clear downregulation of those phagocytosis regulators in the female macrophages with age. And in the males, it's actually non-significant, but if anything, they're trending up. Suggesting something's going to happen to the phagocytic capacity of macrophages with age in females, but this will not be happening in males. So phagocytosis is something we can easily test. And so we're doing this using a fluorescent cargo, in this case, green labeled zymosan. We give purified macrophages about an hour, which allows macrophages that are competent for phagocytosis to internalize those bioparticles, and then we don't wait too long, because if we wait too long, those will be destroyed. And so that's about 1 hour. And so we do this by microscopy and flow cytometry. I'm going to show you the microscopy results here. And the idea is we're looking for the fraction of cells that internalize that green cargo. And so when we look at this, consistent with the omic analysis I've just shown you, we see that there's a significant decrease in the phagocytosis capacity of female macrophages with age, which we do not see in the males with age. If anything, the males are trending up, even though it is not significant, suggesting that there is a functional impact of aging in the females specifically. Now, if we go back to those GO terms and we look at other potentially interesting factors, we also noticed a lot of metabolism pathways that are regulated specifically, in this case, males, but not females. But a lot of metabolism pathways altogether were also regulated in a sex-specific fashion. And so you might be wondering why we care about this, because, macrophages. Well, it turns out that the metabolic preference of macrophages has been associated to their phenotype. So-called M1 macrophages, which is more of an in vitro designation, but let's say more pro-inflammatory macrophages tend to favor glycolysis over respiration. And more tissue repair, tissue remodeling macrophages tend to prioritize mitochondrial respiration over glycolysis. And so the metabolic preference of macrophages can tell us something about their function. And so we, again, use different techniques, but I'm showing you here what we've done using the Seahorse platform with the Cohen Lab at USC to measure that glycolysis respiration preference. And the cleanest results were on the glycolysis side. So I'm showing it to you here, where we're measuring the extracellular acidification rate after injection of specific components. And we can deduce the glycolytic capacity using that second step. And so here I'm showing you the data from one cohort. We have 3 cohorts worth of data to show you the dynamics of it. But basically, we do see that in general, the young females in light pink have the lowest extracellular acidification rate of all the groups, suggesting that they have the lowest glycolytic preference. And indeed, when we sort of extract the glycolysis from 3 different cohorts and put it all together, we do see that the female macrophages tend to increase in their preference for glycolysis with age. And again, we don't really see this in the males. And so there is sort of a specific skew to become more glycolytic with age in the female macrophages, which also tends to be associated to a more inflammatory phenotype. And so anytime you see, sex-specific or sex-dimorphic phenotype, you're curious to see where this comes from, right? What is the mechanism driving these changes? And obviously, when you see sex differences at the basal level, those can come from difference in gonadal hormones, right? Females tend to have this estrogen bias, males tend to have the androgen bias. There's also the role of sex chromosome complements, being XX versus XY, and that's something that Ashley's talk earlier today addressed a little bit. But both of those can have, impact. Now, the only one of those that does change with age, because your karyotype would be largely similar. The thing that changes dynamically with age is actually the level of hormones. There is a decrease in estrogens that's quite dramatic, even though in mice it is not as dramatic as in humans. With age, androgens decrease slightly, not to the same level. But we were wondering if maybe the female-specific phenotypes might be due to the loss or actually dramatic decrease of estrogen systemically. And so I'm just going to show you 2 snippets of data. We think that it plays a role. And we basically looked at conditions where we can naturally or artificially reduce estrogens in young female mice and see what that leads to. The first one is just looking at naturally cycling mice, because as mice go through the estrous cycle, there are times in the cycle where they naturally have high estrogen levels and times in the cycle where they have naturally low estrogen levels. And so if we just take a population of mice on any given day, we're going to have mice in those different phases, and we can isolate their macrophages and look at the phenotypes. And indeed, what we can see is that when we compare high estrogen phases of the estrous cycle, so proestrus and estrus, to low estrogen phases of the estrus cycle, like metaestrus and diestrus, we do see a decrease in the phagocytic capacity of those cells. And to give you context, the difference in the relative estrogen levels between those high and low estrogen phases of the estrous cycle is going to be similar in amplitude to what's going on between average young female and average old female. Now, if we do this sort of artificially by performing ovariectomy or sham surgery on the mice and then waiting a month and getting their macrophages, we can also see that removing the gonad, which will altogether remove the bulk of estrogen synthesis, also leads to decreased phagocytosis. And so we do think that the fluctuations in estrogens can promote decreased macrophage phagocytic capacity. I don't have the time to show you for the metabolism, but the metabolism data suggests that the metabolism is not regulated by estrogen loss, or at least not fully. And so we've been curious in seeing if we could use all that omic data we generated to identify potential drivers of those phenotypes outside of estrogen signaling. And so basically, what we've been doing is looking at things that happen only in the female macrophages with age and not in the male. So first, which transcription factor genes are specifically regulated in the females with age? And so we have a number of those, and I'm going to here pull your eye to IRF2, which is high in the young female macrophages, but low in the older ones, and there's not, it's a little bit all over the place for the males. Next, we can look for in the peaks, the regions that are accessible by ATAC-seq, or the peaks that are marked by, sorry about H4, it's H3K4 trimethylation by cut and run, and look at the peaks that are specifically remodeled with age only in the female macrophages, but not the male macrophages, and look for motif enrichment. Here, I'm just going to show you the ATAC-seq, but we have similar data on the cut and run. And again, one of the top enriched motifs specifically in the peaks that are remodeled only in the females is IRF2. And then when we go into our single-cell RNA-seq data and we look for transcription factor regulon activity change with age using a package called CNIC, we also see a specific decrease in the IRF2 regulon in the female macrophages with age, and there is a small decrease, but it's not nearly the same amplitude in the male macrophages. And so this tells us IRF2 might be playing a role here. And so right now, Ryan is optimizing knocking down IRF2, overexpressing IRF2 to see if we can mimic or rescue the aging phenotypes, but we are pretty optimistic because there is this data set that was generated in bone marrow-derived macrophages from wild-type and IRF2 knockout mice that we've reanalyzed, and the phagocytosis regulators that I mentioned earlier when we analyzed our own RNA-seq seem to be downregulated significantly in the knockout IRF2 macrophages. And then there was another study that looked at the metabolic effect of IRF2, and in that study upon siRNA to IRF2, there is an increase in glycolysis, similar to what we see with aging. And so we believe that this could be sort of a good lead to follow right now and understand exactly what's going on specifically in those female macrophages. And so with that, I've shown you that we have good data that resident macrophages age in a sex-dimorphic manner in mice, that you can use the omics remodeling to predict what's happening functionally to cell populations, that the age-related loss in estrogens could drive aspects of the female-specific functional decline, although not all of it, and that we're now following up on IRF2 as a candidate mediator for some of those other phenotypes that we've seen with macrophage aging. And so before closing, I always like to sort of have a PSA where people keep saying, we want to have personalized medicine, personalized therapies, with the idea that, if you have this SNP or that SNP, we can give you this drug or that drug to improve your chances of responding. But, the biggest genetic differences of them all is a whole chromosome. And so I think it would be very important as a field and, as scientists to start thinking of sex as the first level of personalization of therapies and maybe start personalized medicine by taking into account whether the patients we're trying to do something for are female or male. And so with that, I'd like to thank my fantastic team, all super talented people who've trusted me and work in the lab. I want to highlight Ryan here, fantastic grad student who's spearheaded most of what you've seen today. This work was obviously would not be possible without our funders, specifically here the Pew Foundation. And I want to thank our fantastic collaborators, specifically Helen Goodrich, who's been a fantastic resource on the biology of macrophages and the innate immune system. And I would be happy to take any questions. Thank you.
All right, great, Berenice. Really nice talk. Super exciting. All right, so there's a question that came in on the Q&A, and I'm curious about this too. It's related to so the person who submitted the question is Amin Hagani, and it's related to the use of mice versus humans, given that mice are not, in our mouse facilities, are not generally exposed to pathogens. And so could you comment on the use of mice that are not exposed versus humans in these types of studies?
Yeah, I mean, obviously the end goal is to look at humans. Unfortunately, it is not ethically acceptable to do human knockouts. So for mechanistic studies, we need to use models. And so there's actually some fantastic work being done at NYU, where people are rewilding mice to force them to acquire, wild microbiome and then bringing them back to study them and what happens. And there are big differences. And I agree, that would be really interesting to look at, but we have to start somewhere. And so even though there are limitations, I hope we can learn something important from looking at the mice.
Great. So I have lots of questions. So one question I have, and sorry if you tried this and I missed it, but have you boosted estrogen levels and looked at some of these phenotypes in the old animals?
Yeah, so we've tried this, and the short answer is we don't get any rescue. Pretty much we get partial rescue on one phenotype that I didn't talk too much about, but we don't see rescue. But one of the things that is not very well appreciated by the community is that the ability to respond to estrogen is highly regulated by estrogen. And so the relative level of the 3 estrogen receptors and how they're expressed is affected by estrogen. And the way you respond to estrogen depends on the relative expression of those. And the expression of estrogen receptor alpha is actually one of the top most regulated genes that we see in response to aging in the female macrophages. So we think the only way you could potentially fix it, but this is long-term, so that might be in our future, would be to basically start supplementing just as they enter oestropause. So we would have to start supplementing them when they're 14 to 15 months and then follow them to 20 months. But it's difficult to get approval to give sex steroids to mice for that long because the IACUC's afraid we're going to give them cancer. One of the things we're trying to see is if we can do adoptive transfers. So take the old macrophages and transfer them to young mice to see if we can fix things. And hopefully that can partially answer that question. But yeah, it doesn't look like short-term supplementation in old mouse can fix the problem.
Great, thanks. So yes, I guess it's not that simple, unfortunately. Okay, so a few more questions coming in that I wanted to get to you from the Q&A. Okay, so one question on the activation of gene expression in males versus females. So is there any basic activation which happened early on in macrophages in the females versus the males? Like, I think activation of the macrophages themselves.
Does it happen at a different rate? So the short answer is I don't know. I don't think it's been looked at. Now, in younger animals, like 2-month-ish, there's been some work showing that there is a different rate of self-renewal of those macrophages because they are mitotically competent. There's a different rate of self-renewal in the females and the males. At the age we're looking at, it doesn't look like self-renewal is much of an impact, but we're still looking into it. So I will have a full answer at some point in the future.
All right. So another question from Yusra Ajuao. Great talk. Did you look at other histone modifications? Do you think that epigenetic modifications might be sex dependent or sex specific?
That's a great question. So one of the issues is that when you're looking at naive macrophages from the peritoneal cavity, and I know that's going to make you laugh, Ashley, because it's plenty compared to normal stem cells, but we only get 600,000, 700,000 cells per mouse, at least in the young animals, and we're limited by that, which means that already doing everything we've done is a lot of animals. So we haven't gone to other marks. To answer of whether I think the epigenome in general is going to be sex dimorphic, yes, I believe so, if only because sex hormone receptors are chromatin remodelers. They can kick off nucleosomes and remodel chromatin around them, and also because escapers on the so-called inactivated X include things like KDM6A, which is a histone demethylase, and being expressed at double dose in female cells versus male cells would definitely have impact. So yes, there will be huge differences in the epigenome in general, and I do believe they will be impacted by aging.
Great. So Alessandro Bonetti wants to know whether you considered using nascent RNA-seq instead of the global transcriptome to further pinpoint differences in transcriptional response and efficiency between the sexes.
So again, great question. And I believe that would be really interesting to look at, but nascent RNA-seq work also requires more cells than we can spare. So we would have to look at another niche where there would be more cells.
And second question from the same person, whether you've considered looking at whether female genomes tend to accumulate more double-stranded breaks compared to males.
Excellent questions. We don't know, and we haven't looked yet. But there's so much. If people are interested in double-stranded breaks, I just want more people to look at sex differences. So please look at it.
And another question, sort of related to this this thread. I think you already mostly answered this, whether you've looked at other epigenetic modifications like DNA methylation. Have you looked at that in the different sexes?
We have not, but I know from some publications that people have seen big differences in the DNA methylation of females and males. And actually work from Morgane Levine, who's going to be talking in a couple of days, showed that there is an acceleration of the DNA methylation clock after menopause. So definitely it's very likely there would be differences, but we haven't looked ourselves there.
Great. And then this is from an anonymous person. Is there a senescence deepening that's different between males and females that you know of?
Okay, so this is where we haven't looked at cell senescence itself. I know that I've had some, coffee conversation at conferences with people who've working on senescence, who've told me that the rate at which cells enter senescence and the actual SASP phenotypes are different between genetically male and female cells. So there's likely to be differences, but we haven't looked ourselves, so I can't give any details there.
Yeah, great. And then just more generally in terms of the immune system, are the immune cells functioning differently between males and females?
Yeah, it does look in general, and obviously every time you make a general conclusion, there's probably exceptions somewhere, but it does look that in general, the female immune system is more active and more aggressive in the adaptive branch, but a little bit less, at least in young, healthy females, less in the innate branch, whereas the male immune system tends to be more active on the innate branch and less on the adaptive branch.
Interesting. Okay, a little bit more on the sex-specific macrophage phenotypes. So can the differences observed here be a reflection of an expedited aging process in females, meaning that they will eventually be observed in a much older male?
So that's a very interesting comment. So I think it is possible. And also because one thing that is not necessarily well appreciated is that males actually have quite high levels of estrogens from aromatization of testosterone. So, as testosterone levels go down, you would also have decreased estrogen in males. So anything that's driven by decreased estrogen could also happen eventually in the males. And people who've looked at the phagocytic capacity of macrophages in much older animals, generally in males, do see a decrease. So it is possible that what we're seeing is accelerated aging in the females, whereas the molecular networks behind it are going to be the same or not. I don't know, but that's an interesting thought.
Yeah.
Ashley, I think if you could, Gary has his hand up. If maybe he can open his mic and ask his question.
Sure. Maybe not. Do you want to try again? You can also put it in the chat. Okay.
Yeah, maybe we should just go with
Can it work now?
No, still an echo. I can go ahead and ask. He says, he missed this, but do you think why do you think there's an increase in preference for glycolysis with age? Are age-related effects of mitochondria activity involved, do you think?
So for this, I didn't show the mitochondria data because basically nothing's happening. So we think the mitochondria, at least at the ages we're looking at, are fine and they function fine. It's just really that the cells are shifting to a glycolytic preference. And this is consistent with that M1 phenotype I mentioned, where the macrophages become more pro-inflammatory and start preferring glycolysis. We actually also notice, so there are people who've looked at when you treat macrophages with LPS, so you make them more M1, some genes are upregulated or downregulated. And so when we look at those genes in our RNA-seq, the female macrophages are very clearly shifting to a more M1 phenotype and not an M2 phenotype, whereas the male macrophages with age don't have that shift. And so it's likely that the female macrophages are just experiencing a more inflammatory state with age and the males are not. And that would be the reason they shift their metabolism, not because mitochondria becomes effective.
That's interesting. There's one more in the Q&A. Do you think that the sex differences in macrophages might be tissue specific? Like if you look in different places?
So this is actually a project that we have a fantastic computational undergrad student looking at using publicly available data and data we've generated. And I can tell you, yes, they're going to be extremely tissue specific. The aging part, not as clear because that data does not exist, but the sex differences at basal are going to be very tissue specific for macrophages.
All right. Great. So do we want to move on to the panel? That was fantastic. Thanks for the great talk and discussion. Really interesting.
Thanks, Ashley. So yeah, we want to open it to a roundtable with all the speakers today. So we can take some of the questions that we didn't have time to address before, but I also want to open the floor if anyone in the audience has general questions that they want the entire panel to address, please put them in and we'll go to it. So
Berenice, if you're willing, I might try to have them raise their hand and I might unmute them.
Oh yeah, that's fantastic. Yeah,
yeah,
let's do
we'll
that.
see if that works, and please everybody be kind and
So let's see, I just went back in time while we're waiting for more general questions. So I think this one was for you, Ashley. In aged neurons, so that was from Xiaomin Bao. In aged neurons, do you know if exist binds to additional chromosomes in addition to the X chromosome? And how much do you think the change gene expression you've observed with age is due to neuron degeneration versus just learning? And also great talk, thanks.
Right. Thanks. Good question. So we haven't seen any evidence so far of exist sort of leaking over into other other chromosomes, either the other X that's active or autosomes. But I think, you know, it's when you don't see something, you can't say for sure. So we're actually going in with more sensitive methods to look at that a little bit further. But so far, we don't see evidence of that. And what was the second question?
It was on whether you thought it was neuron degeneration or the fact that the mice have learned over their life.
Oh, that's interesting. So, I mean, we don't know the answer to that, but it could certainly be, I think it would be really interesting to look at whether the activation of the neuron itself and activity over time actually increases this phenomenon, because that is associated with aging, you know, sort of general increased activity in some cases. And so that could be, though that's kind of hand wavy at this point. In terms of neurodegeneration itself, it may be something that makes cells more susceptible to neurodegeneration, but because these mice don't have neurodegeneration, they're just undergoing normal aging. Our model isn't really a neurodegeneration model itself. It's more of an aging model.
Yeah, I'm actually wondering, because I know some people do this, right? Like, if you would have differences in the effect if you looked at mice that had been used in cognitive assays, actually, right? Because you would have taught them things. But yeah, thanks, Ashley. So we also have one for Raul from Marta Kovacheva saying, "Hi, Raul, thanks for the great talk. I was wondering, and sorry if you mentioned it, if you have an idea what the +4 quiescent cells specifically, but not the IGR5 stem cells, would benefit from activating the glycolytic switch."
Well, they are definitely glycolytic, and I heard a question that's one of the reviewers really gave a very hard time to us because they wanted us to specifically delete or inhibit glycolysis in the +4, which was not an easy thing to do. It would have taken us another year and a half for using particular genetic models. So I would imagine that it is unique to these cells because that's where we see the glycolytic switch happening. We don't see it in the LGR5. So I will imagine that inhibiting glycolysis specifically in this +4 will benefit the animals. We haven't done that. We treated the animals with dichloroacetate and the tumors went away. But this was treatment in general, and it was a criticism from the reviewer that says you didn't prove completely with the treatment with DCA that this was the +4 because you are treating the whole animal, but we couldn't do it other way. Carlos, the former postdoc that is in Barcelona, he's now making an animal with a specific diphtheria toxin system to delete the +4, and hopefully this will answer the question. But yeah, it's a good point. I believe that it is the unique adaptation in these cells that is driving the phenotype, but we didn't genetically test it. Thanks.
Thank you. I'm trying to, because it's not always clear to see which question is for who. Do we have any big questions? Oh, there is a question to the panel. I don't know if anyone is going to want to throw their opinion in there, but by Matt Soldano saying, "Great talks by everyone. Could the panelists discuss their opinion on the most recent Dr. Sinclair paper, loss of epigenetic information as a cause of mammalian aging?"
I think Vadim is also, but I am a co-author in the paper. Yeah, I know it's been getting a lot of attention from everywhere. It's a unique study showing, which claiming that when you do a very short pulse of DNA damage, factors from chromatin move to the sites of breaks, try to repair, and the problem is that a lot of these factors, according to David, don't go back to their places where they normally were. So the animal remains with what he called epigenetic scars, areas where chromatin change forever. So independent of the DNA damage. the animals start aging because of these changes in epigenetics. I think the study showed clear changes in epigenetics. And David and the lab went for 10 years. So the long study, they did a lot of experiments to show by these animals are aged around 7-8 months of age that they are looking with accelerated aging. It's clear that a lot of the tissues are exhibiting chromatin changes. The argument that some people were making is, how do you completely prove that this is just not a scar from the original DNA damage, that cells experience the damage, were either killed or adapted to respond to the DNA damage and carry those adaptations from the DNA damage with time. This is a very difficult point to prove. I don't know you can really separate. So it will be an argument that probably we'll be hearing over time when people will be arguing is really a purely epigenetic scar or related to DNA damage that they experienced early by the model he used. But there are clear epigenetic changes, and the paper include one last figure where he tried to reprogram the cells in the eye model that he published before, and show that when he created damage in the eye, he can rescue some of this by expressing these epigenetic reprogramming factors, suggesting that indeed some of the epigenetic changes could be reprogrammed and the neurons are improved. I don't think he's showing, even if the data includes improvement of vision, but definitely improvement in the axon degeneration. So it is an interesting study and suggesting that long-term epigenetic changes could be part of an aging phenotype.
Any other thoughts? Yeah, thank you, Raul, for chiming in. I think Gary has his end up.
I do. Thank you, Berenice, and thanks first to all the panelists. The talks have been excellent. I do have a question for Ashley regarding your talk, Ashley. I wondered if you had planned to look at diet or the use of senolytics in your future studies with your mice populations.
Yeah, I think both of those approaches would be really, really interesting. We have done a little bit of work. We haven't analyzed the data yet using differences in diet, so mostly a high-fat diet, but I think caloric restriction would be equally interesting, specifically looking at the hypothalamus, specific changes in which types of neurons are being modulated in response to those diets. I think that's a great suggestion. We're not quite there yet, but we're sort of just getting started on some of that. I think it could be very important and insightful.
Yes, I was just wondering if there's any senolytics that you're planning to maybe add to the diets.
Yeah, we haven't planned for any specific senolytics, but I'd certainly be open to collaborating with others on this. Those individuals who are doing a lot of senolytic work, please reach out. I'd be happy to work on that. Yeah, it's more a matter of, we just have so many things we want to do, yeah, and so we can't take on everything. But it's not because it's not interesting. I think it'd be great.
Okay, thank you.
Thanks for the question.
Thank you, Gary. We have another question to the panel. The thoughts of people on the panel by an anonymous viewer. So what are the thoughts of the panel on the role of the TET demethylation pathway and the hypomethylation associated with aging? For example, TET2, TET3 contribute to macrophage activation, and their activity is activated by ROS. So again, we didn't look at DNA methylation. So for the macrophage aspect, I'm just going to recuse myself here. But for those of you who've looked at DNA methylation, any thoughts from the panel?
Well, we just haven't looked at it yet. Unfortunately, it's on our list. Super important, but yeah, I don't have an answer for that today, unfortunately.
Yeah, like that's the thing, is there's so many things we wish we could look at, but we
Right.
don't have time. Okay, another question for the panel from Alexei Onufriv. Yeah, sorry if I just butcher your name. imagine a thought experiment in which the organism's DNA were kept completely intact while the epigenetic changes were allowed to happen as they normally would. Would the organism age in this case? So I think this is very akin to asking us about the study from Professor Sinclair's lab in just another way. I think, you know, the thought experiment can be partially looked at in non-vertebrate model organisms because there's been a lot of work just modifying the expression level of histone modification enzymes in invertebrates like flies and worms. And we do know that disturbing the epigenetic landscape or even yeast can modify aging. So this is, you know, as a thought experiment, we already know that messing with the epigenome can lead to aging or can delay aging. So for that, I would say there's definitely some impact here. But, you know, for the rest, that would be basically finding a good model for this, which can be the ice mouse or not, depending on who you subscribe to. Do we have any thoughts from Dow, Raul, or Ashley on this question?
I think that's a great point. And I would just add that not only do we know that modulation of the epigenetic modifiers can alter the aging process, we know that we can rejuvenate cells through reprogramming the epigenome as well. So it's like we know it in both directions. So I think we have pretty good evidence for this. Joe, were you going to say something?
Yeah, so I also want to add that in terms of what we study about nuclear autophagy in senescence and aging, and so we don't think we are screwing up the DNA. And then in this case, you specifically affect the chromatin modifying proteins, for example, the lamin and the WSTF and the other components we have identified. Now, I would also add that the one really strong evidence is from the yeast. For example, Schelleberger's lab have shown that the alteration of the H4 K16 acetyl has a very major impact on replicative lifespan on yeast. The other study, which can probably directly answer your question, is that in the yeast replicative senescence or aging, you have a massive loss of the major histones, 2A, 2B, 3, and 4. Right now, this is from Jessica Tyler's lab, in which they can restore the expression of histone, and then this is associated with dramatic increase of replicative lifespan of the yeast. Right, so I think this will argue that the epigenome alteration is directly causal to the aging process, at least in the yeast.
Okay. Thank you, Dow. Let's see.
Can I ask you a question, Berenice, that I saw in
of course.
the Q&A that I also want to hear your answer to? It's regarding the like going back in the lineage and whether you yourself, or if you know of anyone who's looked at the hematopoietic origins of the sex differences, like have you looked at HSCs? And do you think that could be an origin of these sex differences?
Yeah, so that's going to be a multi-part answer. So the first one is that the macrophages we're looking at, the peritoneal macrophages, are embryonically deposited. So they're as old as you are. So for those that, you know, it might be from the liver progenitors, you know, during embryonic development, but it would be difficult to look at. What I can tell you is a lot of what we see in the the peritoneal macrophages, we also see in BMDMs, which are just derived from monocytes, you know, from adult mice, like we just derive them. And we do see those effects as well, suggesting that the more progenitor compartment already has some effects on there. We've had a collaborative study with Helen Goodridge's lab on just the monocyte compartment, where there are some differences. They're not as strong. I do not know of other studies that have looked like earlier in the lineage. But if I had to make a speculation, my guess is a lot of things might be preset but not visible until you differentiate the cells. Just because the monocytes have lower differences than the macrophages when you differentiate them, that would suggest to me that some of the things might actually be epigenetically set, but not expressing themselves until you force the cells through differentiation. The one thing I want to say, because this is something we've gotten on another paper where we were working on neutrophils, I am not saying that the differences occur at that stage and didn't exist before. I'm thinking that we are interested in seeing where functionally they're likely to have the most impact on the organism's health, though we're looking at the differentiated cells. But it's completely possible that they're set much earlier. And that would, that would still lead to those differences. We're not making a statement on how early those occurred.
And I can add to Berenice, you may remember Jenny Lee's paper on the exist depletion. The main phenotype they saw was tumorigenesis in the hematological cyst, that it was leukemias, suggesting that probably early on the differentiation problem could explain the phenotype. So it's clearly that this may be happening very early during the process of HSC differentiation.
Yeah. Thank you, Raul. Let's see. Well, there's more questions. I see there's a bunch for you, Ashley. So let's ask this one from Chet Lowe, who was wondering if you saw any other genes that are usually silenced after XCI increasing with age, and if exist induction, I guess you did answer that partially, might be a mechanism to silence their genes, or that this induction is actually bad.
Yeah, so it sounds like it's a good question. It sounds like they're referring to genes that escape exon activation. And it's interesting. So we've looked at those and we see them going either direction. So most of them change, but there wasn't as much of an obvious pattern in those. And we think two things. So it may have something to do with like the dysregulation in general and why exist is trying to come up in order to, get all of that back in order, let's say. But then I think the other issue here is that right now, because we only have 2 time points, and I know there was a question in there about an intermediate time point, at least one as well. I think it is important to sort of get a handle on the trajectory of all of this, because when you only have the 2 time points so far apart, then you can get a lot of things changing in the meantime. And so we didn't see such an obvious correlation, but we did look for that.
So it looks like. Oh, sorry, continue. I was going to say, it looks like there's a question in the audience, so when you're ready.
Sorry, I just wanted to ask like sort of a corollary. Did you see any relationship between the distance from the XCI controlling locus and the changes? Is there any effect of how far they are from the locus and their regulation?
I don't think we did that. I'm trying to remember, because I know that's come up in our discussions, but in the end, I'm not sure we did that analysis. I mean, another issue in the mouse is that there aren't as many genes that escape exon activation as there are in the human, and they're also tissue specific. So it's a relatively small number of genes in the mouse that escape in the first place. So it's also hard to really come up with general statements about what's happening. But I don't think we did that analysis. That's a good idea.
Thank you, Ashley. Sorry, Mason. Go ahead.
So there was a question from Prasan Data, and I just unmuted him. So Prasan, if you can unmute yourself and ask your question, that'd be great.
I might have missed it, but I was wondering if there's any role of the dysregulation of NAD metabolism in aging.
I mean, there's definitely a lot of work on that. I just don't think most of us are working on it. Maybe Raul, since T6 is NAD dependent.
I couldn't hear Prasan's question. Can you repeat it, Berenice?
My question was: is there any role of the dysregulation of NAD metabolism in aging?
Any role in what?
Dysregulation of the NAD metabolism with aging.
Well, yeah, why, if you ask, I don't work on aging. I kept saying that. So, but definitely many people working on aging have been looking at the role of NAD. There is clear decrease in circulating NAD that has been shown in many species. And there is a lot of work done with giving animals precursors of NAD like NMN or NR, and that shows improving of metabolic functions, and in some cases, directly aging. I refer you to the work of Charlie Brenner for NR and Johan Auerks, and the work of Shinichiro Imai. Particularly, I think David Sinclair may have also some work, but definitely Shinichiro Imai for work on NMN and some of the NMN-related enzymes and their role in aging. So definitely a lot of work out there on this regulation of NAD during aging and how people are trying to improve it to improve health span and life.
Thank you.
The other thing I want to add is that there is also connection directly to senescence, because it's been reported that the senescent cells contributes to the upregulation of CD38 during aging. So the idea is that CD38 use a lot of the NAD, so then it contributes to NAD decline. So then the CD38 knockout mice have higher NAD level, and then they have better health span during aging. So it's also been reported that the senescent cells through the SAS program will turn on the CD38 expressing macrophages. So then once you have the senescent cells through the SAS, they turn on the CD38 program from the macrophages that further contributes to CD38 upregulation in senescence and the NAD decline. So that I think this is still an ongoing debate. You know, what is the relationship? What is more upstream or downstream comparing senescence versus the NAD? So that study, I believe, is still ongoing.
Thank you, Zhixin. Okay, so also to the audience again, if you have questions for the panel, please send them in. Or if specific members of the panel is fine too, but please say who the question is for in that case. There's actually one specifically for Raul. An anonymous viewer is asking if you think that the epigenetic modifications that cause metastasis are adaptive to the new niche, or if they're already present in the primary tumor.
Yeah, that's the $1 million. 1 million today with inflation, the $6 million question. A lot of work that people has done in the arena for metastatic adaptation. When you look back, they claim that some of these, if you look by single cell analysis, you could see cells expressing. There may be very rare clones that are present, and they are the ones that are adapted. Others will claim that they don't see enough of these changes in the original, and it's really new adaptations. These have been done mostly for genetic changes. Much less has been done for epigenetic changes. And it's a question of threshold. What you will be on a primary, you will be looking for expression of GSDT1, and in bulk, we don't see really, you see very, very little to no expression. But I'm sure that if we look at single cell, we will see some cells rare that may have enough expression that you will consider it positive or high for GSDT1, whether these are selected for later or this is a new adaptation, the cell reach there and they start expressing GSDT1. I don't think we have a way to tell at this moment with the technology available. We probably could do some tracing experiments, but I can't really tell. Which is clear. What is clear that in bulk, at least in these pools, we don't see expression. And if you inactivate GSDT1, we don't see any change at all for the primary tumors. So it's clear that there is no reason for GSDT1 to be expressed in these early lesions because we don't see any advantage indeed. And if we take it out, the primary tumors grow perfectly fine. So if there is any expression up front, it's rare and not required. And they clearly become required and it's necessary when these tumors are in their metastatic niche.
Thank you, Raul. So there's a question that I think we should address. I'm going to use my moderator privileges to bring it to the front. which I think is addressed to you, Ashley. Ali Usel is asking, you use machine learning to measure brain aging in females and found that exists as a significant contributor to female brain aging. So with this information, what do you think of the accuracy or relevance of other brain transcriptional clocks that do not take into account gender?
Yeah, so it's a great question, and if wouldn't necessarily consider our approach exactly a clock yet, but potentially. And so I think we will know the answer to that as more data sets come out, and we learn to to what extent we can actually apply clocks that have been developed in males to females, and then kind of do like the cross validation, right? So I don't think we know the answer to that, and I think that there are certainly going to be sex-specific features that are going to come out, like exist, and I think there are going to be others that are going to come out in the coming months and years, and I hope that that will give us important and new insight into the differences in aging and age-associated disease. So I think it's something that people are excited about doing. I know Berenice is excited about it, and others as well. So so stay tuned on that, I think.
Yeah, and I will just add, I didn't have time to present it because I didn't want to bog down a giant data dump, but we've been doing some of that machine learning on our peritoneal data. And because we didn't want people to say, oh, it's just because you have genes on the X and Y, we actually excluded genes on the X and Y and trained the transcriptional clock with giant quotes. And it is very clear that you need a different one for the female and male cells, even when you don't take the X and Y genes into account. So hopefully that answers that. Okay, there's a question for you, Dao, asking from an anonymous viewer who asked if you think the degradation of WSTF is a result of senescence or a cause.
Yeah, I got it. So the question is the degradation of WSTF is a cause or it's a consequence of senescence. So I think this is a complicated question, so I can probably use an easier analogy to answer this in terms of lamin B1. Right? So we know the lamin B1 is lost at both protein and the transcription level. And then in this case, I think it's both. It's both causal and it's also a consequence. Now, the reason why I said that is because so the results are if you force the cells to lose lamin B1, then even in the proliferating young cells, you can drive the cells to synapse. And so that is already tell you it's a cause. But on the other hand, we do think it's a causal because a lot of times in terms of replicative senescence, you do not lose lamin B1 in quiescence or starvation or other process. You specifically lose the lamin B1 in senescence. Right? So this is going to tell you this is also a consequence. Now, in terms of the WSTF, I think is also the cell line and the tissue and the context dependent, because we know that the loss of WSTF drives the SAT, right? So and it's also known that depending on what context, so SAT sometimes reinforces senescence and sometimes it doesn't do anything, and it doesn't have anything to do with the cell cycle arrest, right? So I think the answer to this question is going to be complicated. Now, for some of the substrate of nuclear autophagy, it's both a cause and a consequence. And in terms of the WSTF, it's very likely to be both.
Thank you, Dao. Someone mentioned as a comment that it might be worth considering Klinefelter males who are 47 XXY, approximately 1 out of 600 males. And I do believe some people try to recruit Klinefelter patients to understand the impact of sex chromosomes. So that is definitely something that's being done. So I think we have 5 minutes left. So let's see, any other questions before we close? Okay, I think there's one for me that just came in. So do I think that the changes I see in macrophages could have been seen at the HSC? And if so, do I think the lineage commitment has sex dimorphism? Yeah, so I'm sorry if I didn't mention that clearly. The macrophages we're looking at are embryonically derived. And so they are the same age. They are not derived from HSCs. They are derived from fetal liver progenitors, and they are the same age as the animal that has them. But there's no reason to believe the HSCs themselves wouldn't have sex differences. And Raul, earlier when I answered the question, also brought up the fact that messing with exist messes up the HSC lineage. So it's very likely that there would be differences, but we haven't looked. And let's see.
Can I ask one of them that I was interested in?
Of course, go for it, Mason.
It was from an anonymous question as well, and I think it was during Raul's talk, but I think it applies to everybody here. And their question was, in general, about histone modifications and why are people using histone methylation and acetylation as the main marks that they're looking at, or the main modifications that they're looking at, and what is the constriction from focusing on other types of post-translational modifications?
Yeah, I can answer that. It's a purely historical reasons. The antibodies from several companies, I'm not saying any in particular, were actually working better for acetyl and methyl groups. So it was easier for people to focus on them. And it's actually funny that even the bivalent, you're probably familiar with the bivalent domains in ESs that Brad Bernstein made famous. They were bivalent for these two marks, the repressive and the activating one, methyl and acetyl. I'm sorry, H4K methyl, 3K4 methyl, and 3K27 methyl. It's just that the antibodies work better for these marks. We now know that there are many other modifications that are beyond acetyl and methyl, and with much better antibodies. It's just that people historically are so used to use those. They know that they are correlated with activation and repression, so it's easy to use the one that are known. There is no real restriction biologically to suggest that they will be more important than, say, A3K, some of the phosphorylation marks, some lactylation. You probably are familiar with the beautiful recent paper, last year paper from Yingmin Zhao on histone lactylation with major roles in changing transcription. So again, the main reason was historically, and if you are working on other modifications, don't feel limited because of what is being published on acetylation and methylation.
Yeah, I can say the only reason we're using H3K4 trimethylation is that we know there's a great antibody that is very specific, and we know where we expect to see it and what it's supposed to do. That's the only reason.
Yeah, it's very tool driven to some extent, but more antibodies that come out that are very specific, the more ability we have to branch out and look at other things, like all the marks that Raul mentioned. That'd be great.
Yeah, Gary?
Hey again. Yeah, I have one other question. I noticed that some of the work is done on mouse models. I was wondering if you guys have any collaborations or expect to, or are looking for collaborations with groups that are looking at more long-lived species, anything from primates through to, I don't know, even elephants, right? To see if those changes with age hold in these other models.
Yeah, I see that Vera left. Definitely Vera would be the more appropriate person to answer. She's been working on all the long-lived species and published on them, a very unique set of fibroblasts that she sent to one of her students to Alaska to get from the bow whales. And a lot of this work, she has been shown correlation with a longer living species, but I don't know if anyone else has experience.
We have
I
was going to say we have a new group through this NSF funded center that I mentioned, and we're actually going kind of the other direction. So looking across evolution at a variety of species, some flies, some wild species, butterflies and bats, and it's an NSF-funded center called Ice Age. So, and we're looking at sex differences. So I think give us a year or two, and I think we'll have some cool data. But we don't have any of these like really long-lived species like what Vera is looking at. I think that's going to be really exciting as well.
Yeah. Okay. Thanks.
Okay. Well, I think it is 11. So thank you all the panelists for great talks and stimulating roundtable. I hope everyone had a great morning, and I hope we'll see everyone tomorrow.
Thank you, everybody. From everyone at Motif, we're really grateful for all the presenters and all the talks. Excellent work. Thank you.
Thank you.
We look forward to seeing most of you tomorrow. Thank you.
Yeah, for the audience, please join us. Our next session will be tomorrow and again on Wednesday. So hope to see you there.
Bye.